Well, welcome back. It's my great pleasure to chair this session. And as always with these sessions, my head is already full and we've only just really started. This session is going to drill down on, I think, a key issue that emerged from the previous session, namely interdisciplinary approaches to measurements and models. And we're going to split the session into two halves with lunch intervening. And it's my great pleasure to introduce Professor John Gallagher from Oxford University. So John is the Professor of Cognitive Health at Oxford University and crucially Director of Dementia's Platform UK, DP UK, which we've already heard refer to in session 1. So John, if you're ready, over to you.
Okay, thank you very much, Carl. So let's start at the beginning. Beginnings are such delicate things. My name is John. I'm Director of the Dementia's Platform UK. And the work I'm going to present is really the fruit of many people's work. And several people in the meeting today have contributed to it, so I can take very little credit for it, really. I mean, Paul Matthews and Craig contributed to the thinking and to DPUK in the past substantially. And Ivan, who will be speaking later, is also part of it. James, Ivan leads our Recontact Register work, and James Rowe leads our experimental medicine work. But we are a fairly complex organization, and I'm really going to focus largely on the data portal today, just through the sake of time. Let's just frame the data integration challenge. So we live in an age of increasing quantity of data. I think exponential growth is probably an understatement. The data in relation to health are increasingly complex. It's not just what affects the brain, it's what affects the heart, it's what affects the gut, and they all interact, surprise, surprise. There is increasing technological diversity. So it's no longer sufficient just to be an expert geneticist, you have to be an expert imager as well. There is increasing public sensitivity because with all of these very detailed measures, there is the increasing risk of identification and all the social consequences that go with that. And that really boils down to increasing inaccessibility for science. So there's an unusual conundrum here. We have more data, it's more informative, it's more interesting. Actually, it's less accessible because to make the most of these opportunities, you need competent systems and competent individuals. And this, well, perhaps it's unsurprising, but this is a major challenge. So in DPUK, we've attempted to simplify the problem. Although it's Dementia's Platform UK, we really have tried very hard to develop this as a generic solution. So we're trying to develop what we would describe as a high trust system, which is transparent, secure, and fully auditable. We are trying to streamline our data access routes through standardized legals, approvals, and analysis environments. We want to standardize the data models in terms of the data structures and the convention used to describe them. And we want to work towards building integrated networks. So we want the DPUK to be interoperable with other platforms to allow integration. And this means integration across modalities, but also integration across jurisdictions. So I'm going to look at all of these issues, but not in a linear form. I'm going to describe the data platform, and these various issues will be alluded to as we go through. So here we have the overall view, the overview of Dementia's Platform UK. We have the data portal, we have a trials delivery framework, clinical studies register, and we have experimental medicine incubator. And these are in the context of imaging data, stem cell data, informatics. So you can see that there is a very strong case for addressing these issues within DPUK. So let's look at the data portal then. And the data portal provides a single point of contact, and that allows us to standardize because effectively the environment is under our control. So we can go to experts in the field, we can consult, but nevertheless, it is within our gift to say, okay, these are the ways that everybody else does it. Now, this is a way we can take the best from all of these different approaches and create something which most people, most of the time, will find helpful. So we standardize data curation, which is actually a fairly challenging and thankless task, but I hope you'll realize that it's very worthwhile. We can streamline legal and access procedures. If you go to any university, its legal department will have its own way of doing things. And if you want to access data from several institutions, it can be extraordinarily difficult. And we are developing centralized linkage to other platforms, such as NHS Digital and the Alzheimer's Disease Data Initiative. We have created a flexible and scalable workspace. We have a suite of data discovery tools. I think they could be improved, if I'm honest with you. We can conduct pooled, merged, and pooled and federated analyses, and we have an environment which can cope with, the more or less the entire breadth of analytical techniques. Although with some heavy compute issues, sometimes you need to talk to us about getting a capacity given to your workspace. It has a multimodal capacity, which I'll come along to later, and it is a hybrid cloud architecture, so you can do on-premise as well as in-cloud analyses. And of course, that boils down, that's an economic issue, really, largely. The cloud might be wonderful in very many ways, but if you're storing data there for large periods of time, it's certainly not cheap. And we can configure it for individual workspaces and for consortia workspaces. So the data portal itself is designed as a knowledge environment to allow these functionalities that I've described. So we can ingest multiple data sources, bring them into a single location, virtual location. We can then curate these data to a standard. That's, as I said, very labor intensive, but we're working on it. We have to construct a data model, which I'll describe later. We have to work through prioritized data sets, et cetera, et cetera. We need to be able to talk to other platforms, and the data need to be discoverable through the different tools. We need to access broker with the data controllers, because DPK acts exclusively as a data processor. We do not own data. We can take no credit for the data. The credit for the data lies with the people who designed and collected the data in the first place. But we do provide personal workspaces for data analysis so that you can access multiple data sets through one login. And then we are developing a knowledge management system so that you can rapidly, third parties can rapidly access data sets and data code to replicate analyses and take the field forward. So if we come on to the data analysis pipeline, this is where the multimodality really comes into its own. Multimodal data is very, very complex. If you get it in its raw or native form, you might have DICOM or NIFTY files, as Heidi has described. And she also described how the Oxford group have produced pre-processing pipelines to arrive at image-derived phenotypes. That is absolutely incredible. That's transformative. That is absolutely transformative. And we may hear from others later how there are pipelines being developed for the transcriptome and proteome metabolome so that we can arrive at omics phenotypes. For genetics, the pipelines are reasonably well agreed in principle, but standardizing them is very difficult. Electronic health records, the pipelines, well, I'm yet to be convinced that they really work, if I'm honest with you. For digital devices, there are no standards. It's a bit like the Wild West, really. So it's very hard to have pre-processed data that the non-expert can access. For the survey data, which as an epidemiologist I'm more familiar with, every study employs its own data model, and there's very little rhyme or reason as to how they may be compared. And this is understandable. I'm not being hypercritical here, but if you're doing a longitudinal study over 30 years, you inherit data practices which are 30 years old. And those data practices were cutting edge then, but they may not be considered to be cutting edge now. So what we're looking to do, and we're part of the way through, is taking raw uploaded data, which we would take from any source. We then seek to pre-process it so that it can be made accessible to the non-specialist. Now, I don't mean the non-rigorous scientist, but what I'm trying to get across is that you don't have to be an expert geneticist to use genetics data from within the data portal. You don't have to be an expert imager to use image data from within the data portal. We would like to bring them all together so that we can conduct, so that in one sense we can, how can I put this, make available to all, you know, whether you are in Namibia, whether you are in Australia, whether you are in Manchester, you can come to a single place, access state-of-the-art, cutting-edge data in a rigorous way, which allows you to test your very, very best ideas. So that's what we're trying to do. And this is the, if you like, the model that we're using of taking the raw data, standardizing all the agreements, pre-processing it wherever we can so that you can access it more easily. So does it work? Does it work? Well, we've created this secure environment, which I described previously. And we find that by and large, data owners or data controllers are reasonably willing to trust this environment. And trust is the very big thing here. So they're reasonably willing to trust it. And we are very flexible in the way that we make agreements with them, in the way that we receive the data. We entirely respect their data access procedures. We encourage them to adopt ours because ours is streamlined and easier. And you know, a fair proportion do. But what it means is that we can facilitate data access. And I use the term facilitate because you know it boils down to an agreement between the researcher and the data access committee of that particular data set. We can facilitate data access for about 42 cohorts with, well, around 3.4 million individuals. So of these 42 cohorts, 90% accept our standard legal agreements, which is very, very encouraging because it just makes life easier for everyone, for the universities, for the researchers, for us. 67% accept a streamlined data access process where we have a standard data access application form. It covers all the bases that those data access committees require. And again, people know what they're getting. There's nothing, they don't have to read it through 10 times to understand it. They know the structure, or they know what they're looking for, and they can turn it around rapidly. And 52% accept grouped data selection. And this is a very important point. If you're going through a study of 4,000, which has 4,000 variables measured on each of 4 occasions, it's much easier just to say, please, can I have access to the cognitive variables? Please, can I have access to the dementia variables? And rather than I want this individual variable, that individual variable, then you have to come back to the later date and say, oh, I forgot this one. Can I have this one also? So we're working quite hard in a sort of fairly nudge, gentle encouragement way for the cohorts to accept grouped data selection. So over the last 2 years, we haven't been going long, maybe 2.5 years since we launched the data portal. We've had 228 proposals. Now, these proposals largely are multi-cohort proposals, because that boils down to 845 cohort access applications. Of these 228, 61 failed triage. Now we do triage them because some of them just are very simple. They say, give me all your data. And this doesn't go down well with the data controllers, and it does betray a lack of thought on the part of the applicant. So we triage those out. But we've had applications from 404 applicants, nearly 100 institutions from 22 countries. Of the 845 applications, 522 were forwarded to the data access committees. Half of them were approved. A quarter were declined because, of those, 16% were due to not asking for the right data. I think the interesting issue here is that we had a decision from the Data Access Committee rapidly. So the median time was 23 days and the mean time was 39 days. That is remarkable. If you've ever tried to get a data access decision, it usually takes several months. This really does largely boil down to the development of trust between DPUK and the Data Access Committee and the use of standard procedures. So I'm going to talk about survey data now, the curation of survey data. If you go to these cohorts, as I said earlier, everyone will have its own data model. And if you want to look at ELSA data, Generation Scotland data, Memento data, you have to learn 3 different data models before you can even begin to understand what is there. And then this happens for every time a researcher wants to go to look at a new data set. It's a lot of redundant activity. So we've looked at the data access requests that we've received. We've consulted with data expert epidemiologists, and we've realized that you can really, from a data discovery perspective, because I think every data structure is purpose-driven. From a data discovery perspective, basically you can break it down into about 18 areas of data that people are interested in. And then again, from a data discovery perspective, you can have too much complexity and too little. So it's what seems to be a reasonable level of detail is breaking each of these areas down into 4 levels. So we've looked at cognitive status here. You can look at the theme of cognitive status. You can look at different cognitive domains, and within, say, memory, you can look at different types of memory, going down to the measures that are being made. Now, this is not a data model which allows you to, if you like, have a fully logically consistent breakdown of the data. It's a data model specifically designed to help you identify, and so it's a data discovery curation model. And this has some epidemiological pedigree, and we are working with Gates Ventures and the Alzheimer's disease data initiative to roll this out through multiple cohorts. But the other thing you need in a data model is standard conventions. So we've looked at, we've had talk about time, measurement of data over time, measurement of data in different data sets in different ways. Effectively, our analysis suggests that there are a very few parameters required to uniquely identify a variable or a datum, if you like. There's the data source, what was measured, its chronological context in the terms of was there any repeat measurement for precision? For example, taking blood pressure 3 times and then taking the average of those 3 measurements. And was there repeat measurement for change? Taking blood pressure at recruitment, taking it 5 years later at a follow-up or whenever that might be. Now, with these bits of information, you can then generate a variable name which brings all of this information together in one place. So this is a unique variable name which gives you the cohort, the measurement, the array, and the wave. Now, once you are an analyst, as an analyst, once you have that information, you can change that into any naming system which you prefer. I have my own abbreviations, which I'm very familiar with. But the point is, whenever you come to any particular data set, you then have a standard set of variable names, a standard structure by which you can rapidly understand what the variable is, when it was measured, and from where it came. So where are we going in the future with this? Well, my belief, and if I'm right, you can buy me a pint, is that we are moving towards national networks of trusted research environments. So the Dementia's Platform UK Data Portal is what we would describe as a trusted research environment. It meets all the security requirements that government and the law requires. It allows data controllers to be absolutely sure that they know what's happening to their data. There are no data transfers beyond the research environment. So I would imagine that the UK in the next 5 years will move to a model of trusted research environments. And for example, this one here might be UK Biobank. This might be genomics, GEL. This might be Deep UK. This might be the Essex Data Archive. And these trusted environments will either be on-premise, or I expect there'll be hybrid cloud on-premise solutions. And I expect that they will become, although they're not yet, but they will become interoperable, so that a single login user face will allow you via your personalized workspace to go and bring data from each of these into a central location. I think we have a bit of work. I mean, we operate this at Deep UK, but I think when you go to other TREs, there is a little bit of quite reasonable and important discussion to be had before this is realized. But I think this model will be, in one way or another, broadly realized in the next 5 to 10 years. This model will also is likely to operate in other countries, because the issues of jurisdiction are critically important here. So you can imagine similar trusted research environments, a network of that in, say, Australia, or another one in Canada, perhaps. It's particularly useful where you have integrated health systems. It's not so obvious how it would work in a less integrated health system, say, like the US, but nevertheless, I think this is what is like to happen. And from your single login user interface, your personal workspace, where you have all your bona fides agreed, you should be able not just to talk to different TREs within one jurisdiction, but also TREs between jurisdictions. Now, this is, I think, it is the future. I think there'll be, it's actually highly political. I think that this will require the goodwill of governments. It will require if you like, the desire of scientists and the demonstration of scientific value. But nevertheless, I would be very surprised if this doesn't describe something similar to what we will see in 5 or 10 years' time. Thank you.
Well, thank you, John. That was a fascinating insight into the vexed and multifaceted problems that you're contending with. So I love the distinction between give me all your data and a trusted research network. Wonderful. And you will be available at about 3 o'clock, hopefully, to join the speaker panel Q&A later.
Yes,
on.
yes.
Thank you so much. So it's now my great pleasure to introduce Professor Setterberg, who's spent many years developing biomarkers in Alzheimer's disease and the like from the Queen's Square Institute of Neurology. So Ulrich, over to you when you're ready. And we can see your slides. That's brilliant. Thank you.
Thank you very much. Thanks for inviting me to this exciting day. It's been a pleasure listening to the talks. And I now would like to give you an overview of the latest developments in the fluid biomarker field. I will try to cover some of the CSF biomarkers, but also update you on the recent developments in in blood biomarkers. And then I will have a final slide also indicating what else could be done now. And many of the new biomarkers, they really need to be evaluated in interdisciplinary research, comparing the fluid biomarker results to imaging results, to clinical phenotypes, to follow up, to a lot of different things. So there will be a certain focus now on Alzheimer's disease, and you are all, I think, familiar with this type of revised classification of patients with suspected Alzheimer's disease. So one would do a quite careful workup regarding the clinical symptoms, take a careful medical history, of course, but then the suggestion is to classify individuals according to whether or not they have biomarker evidence of amyloid, tau, and neurodegeneration pathologies. So this has been described in a research framework, but in many places in Europe, it's actually used. It has sort of crept into the clinical work also. At least in Sweden, it's quite quite a lot dependent on the interest of the physician, exactly how he or she operationalizes the diagnostic algorithm. So I work both in the UK DRI, in the fluid biomarker lab there at UCL, and I also work in Gothenburg, Sweden. Now I've been stuck here in Gothenburg for a while, and I so miss going to London. I hope it will soon be possible to start traveling again. So if we talk about the ATN biomarkers, we have the amyloid criterion, that we have CSF beta 42/40 ratio. The 42 amino acid long beta amyloid is selectively depleted from the cerebrospinal fluid if there is ongoing amyloid buildup in the brain tissue. And this does not happen for A beta 40. So a reduced CSF A beta 42/40 ratio is a very well-established biomarker now for amyloid pathology and has high concordance with amyloid PET. So these two biomarkers are sort of established now. For the T criterion, tau, the best biomarker is probably tau PET. You can in CSF you can measure phosphorylated tau forms, which correlate with tau PET signal and also neuropathology, but the correlation is weak, and it looks like CSF phospho-tau increases before tangle pathology is manifest in the tissue. And recent data actually indicate that the way we should interpret the fluid biomarkers for tau is that neurons exposed to amyloid will respond to that pathology by phosphorylating and secreting tau. So to some extent, the fluid tau biomarkers are more amyloid biomarkers than tau biomarkers, to put it perhaps in a bit oversimplified manner, but that's actually what it looks like. For the N criterion, neurodegeneration, we have CSF neurofilament light, which is the best established fluid biomarker for neuroaxonal degeneration, which is positive across neurodegenerative dementias. And then we have, of course, magnetic resonance imaging and FDG PET and other imaging methods. So this is what it looks like in regards to imaging and CSF. But can this be measured in blood? Of course, listening to John's talk and previous talks also, if we had simple blood tests, we could do quite exciting research in big biobanks, and this is where the whole field is going now, and this is where UK is uniquely placed in the world to really lead on this, I think. And the answer to this is actually yes, and I will show you the data supporting this now. So if we start with amyloid, plasma beta amyloid concentrations have been possible to measure for decades, but we have probably not been that good at measuring them. One here indicates no change, and then you can see here that different studies from 1999 and onwards show results on plasma beta levels in Alzheimer's disease control cohorts and also some mild cognitive impairment cohorts, but the results can go anywhere. Some report increased levels, some report decreased levels, and most report no change. This has been the situation for many years. And it actually made me think that the blood test for amyloid would not be possible. But the peptide is so difficult to measure. It's sticky. It binds to a lot of different plasma protein. It's much easier to measure in cerebrospinal fluid. But then in 2017, this paper was published from Ovod and colleagues. This is from the research group led by Randy Bateman in St. Louis, Missouri. So what they did was that they developed an immunoprecipitation method where they could extract all beta amyloid from plasma. Then they took and eluted the amyloid peptides, digested them with trypsin, and measured the C-terminal fragments, by which they could, by mass spectrometry, measure the 42/40 ratio. And then they got this result, where in amyloid PET positive individuals, they see this type of reduction. These are amyloid PET negative individuals. There is an overlap, but longitudinal studies indicate that some of these people actually move to the amyloid PET positive group. So it looks like the ratio goes down a little bit before amyloid PET turns positive. But there is one thing one should observe in this slide, and that is the y-axis. So you see that in cerebrospinal fluid, the reduction of the A-beta 42/40 ratio is 50% with a very small overlap between the two groups. But here you see, if you consider that the y-axis is broken here, that the reduction is around 12, 13, 14% between these groups. And the lower reduction in plasma has a very simple explanation. That is because there is quite a bit of amyloid in plasma, which is blood platelet derived, hepatocyte derived, muscle derived. So the CNS part of the blood signal is smaller in blood than in CSF. This reduction happens on a background of non-relevant beta amyloid. That's one way of interpreting or explaining these results. But still, a signal with an AUC that looks quite good actually. But it's if you run a clinical lab as I do, it's a bit of a nightmare to try to keep this type of assay stable over time. A small percentage reduction that you should be able to detect week by week in a lab, that's hard, but doable, I think, with new technologies. This is another study by that came the year after from a Japanese-Australian collaboration, Nakamura et al. In Nature. They, in two independent cohorts, they measured by mass spec the A-beta 40/42 ratio. They have an inverted ratio because then data get more normally distributed. And then in amyloid PET positive individuals, they see this type of increase. There is an increase because of the inverted ratio. And this is the replication cohort, an increase in the ABLE study from Australia. So basically, I see these two papers, Over et al. And Nakamura et al., as two papers showing essentially the same thing, but with slightly different methods and some different cohorts also. So I'm pretty convinced that this is working. And we developed another method ourselves, which also is mass spec based. So we add recombinant heavy isotope labeled beta amyloid peptides to 250 microliter plasma. We pull out all the beta amyloids using antibodies conjugated to magnetic beads. We dry, elute, and perform HPLC and can thereby measure A beta 38, 40, and 42 with quite good precision, actually. And then we did it on the 1946 cohort, the UK birth cohort, which has this type of cognitive sub-study called the Insight 46 cohort, led by John Schott and colleagues at UCL. And there we can actually see that, I should say that these are cognitively unimpaired. Around 19% were amyloid PET positive. So they have preclinical amyloid pathology, not severe enough or not long-standing enough to have had time to cause cognitive problems. So preclinical amyloid pathology, and that can be detected with a plasma beta test with an AUC of 0.82, which is not that bad, I think. We have also, together with Roche Diagnostics, tried to develop clinical chemistry-like tests because the mass spec-based tests are quite laborious and hard to do in general laboratories. But this is an A-beta 40 and 42 test developed on the COBAS platform, a real workhorse in clinical chemistry labs. So you can take a blood sample and 17 minutes you have the 17 minutes later you have the A-beta 40 and 42 concentrations. And then you can see here that you get a quite nice AUC also for this simplified clinical chemistry test which is both high throughput and high precision. The AUCs are not, if we compare with mass spec, they are a little bit lower, but by themselves you can detect amyloid with 0.8 and 0.8 AUC. And if you add some other variables, you can move up towards 0.86, 87 or so. But here actually ApoE and does give you some additional benefit. But I don't like that approach really, because it's a bit of cheating, I think. ApoE is not a biomarker, it's a genotype. Then if we move to the T criterion, phospho-tau, this can also be measured in plasma with ultra-sensitive measurement technologies like single molecule array. And the first paper on this came in 2017 from a Japanese research group led by Tatibe and colleagues. And what they did was that they took the ultra-sensitive commercially available total tau assay that we have used for 10 years actually on blood samples. It works to detect acute brain injury, but it's not that good for chronic neurodegenerative disease. And then they replaced one of the antibodies with an antibody specific against an epitope consisting of tau phosphorylated at amino acid 181. And it's not a big paper, this one, but I thought it was so nice. It's 15 controls, 28 dementia patients, and the data look like this. The dashed line here indicate a lower limit of quantification. So these are, the assay can't detect all samples, which is a drawback that needed to be resolved. But among the measurable levels, the AD group was clearly overrepresented. And then if one took the measurable phospho-tau concentrations and correlated with CSF, this quite nice positive correlation was revealed. So to me, this was it's a small study, but I thought it was a beautiful study and encouraging. So it really indicated that there might be a phospho-tau signal in plasma. Moving on here, Jeff Dage at Lilly developed a phospho-tau 181 assay on the Mesoscale Discovery platform, which is sort of semi-sensitive. It's not as sensitive as Simoa, but it's around 10-fold as sensitive as regular ELISA. And then he could, together with Michel Mielke, quantify phospho-tau levels, phospho-tau 181 levels in a quite large cohort from the Mayo Clinic. So here are cognitively unimpaired amyloid negative individuals, cognitively unimpaired amyloid positive. There is a small trend towards increase, but not significant. These are patients with mild cognitive impairment who do not have amyloid, mild cognitive impairment patients with amyloid, and either dementia patients with amyloid. So here we start to see a signal. There is some overlap again, but it starts to look promising. And then Jeff refined the assay, and together with this study that came in, was published earlier, well, spring 2020. And here we could see this type of result. Healthy controls, MCI patients, AD patients, clearly elevated. And then for plasma phospho-tau levels. Other tauopathies like PSP do not show increased levels. And this is what it also looks like in cerebrospinal fluid. And of course, it seems a bit counterintuitive if we think plasma phospho-tau or phospho-tau 181 is a tangle marker. But it is actually explained by the fact that it's not really a tangle marker. It is a marker of a neuronal response to amyloid. So again, amyloid exposed neurons will react to this pathology either through microglia or through a direct interaction or a combination thereof, phosphorylate and secrete tau. And that is why it's such an AD-specific test. For these diseases, we need to continue to work on better tau biomarkers in the fluids. This is the correlation between amyloid PET and phospho-tau levels, and here you see tau PET correlation. So neurons that are exposed to amyloid will eventually develop tangle pathology. So there is a type of indirect predictive correlation between phospho-tau 181 and tau PET signal. Not side by side, back to back with the previous publication, this study was published where we could compare phospho-tau levels with tau PET results and sort of tau PET BRAC stage the patients. And you see this type of stepwise increase. So the more tau pathology, the more widespread Alzheimer's disease, the higher the plasma phospho-tau levels. But there is a reaction in amyloid PET positivity in regards to amyloid PET positivity only. It looks like this. This is our own assay that we developed on single molecular array, the single molecular array platform, and that's available also at the UK DRI lab in the Cruciform building in London. So it has a nice lower limit of quantification. It's completely selective for phosphorylated tau, no reaction with non-phosphorylated tau, which is nice. Here we have young controls, cognitively unimpaired controls, MCI patients and AD patients with a clear separation. Actually, it starts to look like performance one would expect for clinical chemistry tests if we think about clinical chemistry tests in medicine in general. And this is another independent cohort where there was also an FTD group. And here you see FTD patients and AD patients can be completely separated with this plasma phospho-tau test. Here's the correlation with tau PET. And here is a correlation also with amyloid PET for plasma phospho-tau. And this is in familial Alzheimer's disease, the UK part of the DIAN cohort. Non-carriers, pre-symptomatic carriers, and symptomatic carriers. And then we have these odd high ones in the non-carrier groups. This might be related to home collections and delayed time to centrifugation. This we didn't know when we published the study, but we have done experiments on this. It looks like if you collect whole blood, you need to put it in you need to spin it and separate the plasma from the cells, not to get in vitro phosphorylation of tau. So that's but I mean that's the regular way you normally handle plasma phospho-tau. So for home collections, one would need to bring a portable centrifuge. This is the data when you separate group level data on plasma phospho-tau levels in relation to expected year of onset, and here it looks like it starts to change around 10 years before expected year of onset in familial Alzheimer's disease. And here is again the Insight 46 cohort. So the plasma phospho-tau's ability to detect preclinical amyloid. The AUC is 0.72, so not optimal, but it's interesting still that there is this type of amyloid detection by a plasma phospho-tau measure. This is in Down individuals. This is with Juan Forte and Andre Stridham also. The London's cohort has very similar results. Down individuals get Alzheimer's disease at have a very high risk of getting Alzheimer's disease. And then it looks like this, that you have asymptomatic, progressive, and dementia Down individuals with this type of stepwise increase. And in relation to neuropathology, we have data together with King's College Brain Bank, where antemortem plasma phospho-tau levels really show that they reflect AD pathology, irrespective of whether or not there is additional pathology. But if it's not AD pathology, the levels are normal. And then this study was published during the summer, phospho-tau 217 for Alzheimer's disease versus other neurodegenerative diseases, where to wrap it up here, phospho-tau 217 looked better than 181 as a phosphoform, and actually looked like it could behave. It worked as well as CSF and tau PET biomarkers, so very promising. So we really have a phospho-tau signal in blood that works quite well now. For neurodegeneration, we have neurofilament light, which can be measured in blood now with a nice correlation between plasma and blood, CSF and blood. This is the familial Alzheimer's disease group with the neurodegeneration marker increasing from pre-symptomatic carriers. They are significantly higher than non-carriers, but then the clear increase is seen in symptomatic carriers. And this is individual data. So black lines are non-mutation carriers, horizontal lines, no neurodegeneration, and then mutation carriers who start to get close to symptom onset, they shoot off in their neurodegeneration marker like this. And of course, one would like to treat them and bring them back down again with a disease-modifying treatment. In ADNI, it looks like this. AD patients have a higher slope, start off higher, and then MCI patients are in the middle and controls here. I will wrap up now so that I don't take too much time here. So for blood, with all the biobanks that are available in the UK, we can now with simple blood tests do ATN classification of the subjects. And of course, this would be so interesting to relate to genetics to clinical phenotypes to a lot of life course, lifestyle related variables. There are so many studies we can do through this. So amyloid, we have the plasma beta-42-40 ratio, tau, plasma phospho-tau, neurodegeneration, plasma neurofilament light. And then GFAP is another marker that shows up now as an astrocytic marker that reflects what's going on in the brain and has a CNS selective expression. There are lots of other biomarkers we can measure. Many of these are easier to measure in cerebrospinal fluid. Astrocytic and microglial markers are often also expressed in the periphery, so here one might need to continue to work with CSF. The elusive TDP-43 pathology, where we have failed to develop both imaging and blood and fluid biomarkers, is still a difficult one, where we need to continue to work on fluid and imaging biomarkers. But actually, a few months ago, an RT-QuIC-like method was published for TDP-43, and I thought it looked super exciting. That you have this idea, this vision that in cerebrospinal fluid you could have seeds of misfolded TDP-43 if there is such pathology, and the same has been established actually for alpha-synuclein. So I think these RT-QuIC-like assays could potentially help out, but then you have to collect CSF at the moment. So to sum up, CSF and plasma biomarkers, they work well to classify patients according to the ATN criteria, which should be useful in clinical trials, genetic and epidemiological studies, as well as in clinical practice, I hope. And perhaps especially important once we get some treatments approved. Then there are many other candidate biomarkers for lysosomal dysfunction, blood-brain barrier dysfunction, microglial activation, astrocytic activation, where there are promising results in cerebrospinal fluid, but more work is needed. NFL is a great marker for general neurodegeneration and works in blood. Phospho-tau looks also good. There are promising results for alpha-synuclein pathology. I should add TDP-43 pathology here also, RT-QuIC and PMCA. You need CSF. And then fluid biomarkers for TDP-43 pathology needs further development. And interdisciplinary work is key, because we can't understand the fluid levels if we can't compare them to neuropathology, neuroimaging, and a lot of clinical phenotypes. Here's my Gothenburg lab, and here is my London lab. Thank you very much.
Well, thank you. That was a really fascinating overview of the field from the point of view of chemistry and neurochemistry, and beautifully illustrating the interdisciplinary aspects which you ended on. So that was great. Thank you so much. And hopefully you will be available later at 3 o'clock to discuss it. Brilliant. Thank you very much. So now we're back to Oxford University Department of Psychiatry with Dr. Ivan Koichev, whose special expertise is wide-ranging, but with a particular focus on experimental medicine and clinical trials. And he's going to talk to us about novel scalable technologies for preclinical detection of dementia. Ivan, over to you when you're ready.
Many thanks, Carl. So yes, my background is very much on the lines of running clinical trials that are experimental. So trying to, for example, detect that a particular compound is hitting its target. And Alzheimer's disease and dementia is a particularly interesting proposition because of this graph. And previous speakers have talked about this at length, so I won't spend too much time on this. But roughly what this should tell you is that the pathological process begins up to 25 years before first symptoms. So this, for example, is data coming from people who have the autosomal dominant mutation. That means that they're essentially destined to develop early onset Alzheimer's disease. And when you follow people up who are essentially the children of patients with this mutation, they have a confirmed mutation. When you follow their children up, knowing that they will develop Alzheimer's disease, you can start to see that already 25 years before first symptoms, you start to see this extinction of amyloid that Henrik was talking about in the CSF. Then about 10 to 15 years, you start to see a rise in the marker of neurodegeneration, which is CSF tau. And then we're starting to seeing a dropping off about 5 to 10 years in glucose metabolism using FDG PET as well as hippocampus. And while that may be quite scary, because I think it really paints a very clear picture, this is a progressive process that takes decades to manifest in the punctuated line, which is where we see people in the clinic with symptoms. Also, it is a huge opportunity because one of the main interpretations of why treatments have failed so far in Alzheimer's disease is that they have been tested in too late a stage. And that's why a lot of the experimental medicine in Alzheimer's disease has shifted towards this preclinical stages. And another thing that has become evident from studies such as these that we ran into US cohorts is that in addition to knowing that this process starts up to 25 years before first symptoms, there also seems to be a rapid inflection, sort of a time point in one's life course when there's a rapid inflection in the rate of accumulation of Alzheimer's disease protein. Here, we demonstrate on 2 different cohorts that there is a time point around one's late 50s to beginning of the 60s when there's a rapid escalation in the tau protein and also rapid loss of amyloid in the cerebrospinal fluid, indicating that this person has entered the more aggressive parts of the Alzheimer's disease trajectory. And that we know that there is a genetic variant, so ApoE4, essentially the E4 allele of the ApoE gene, which makes this accumulation start earlier and also progress faster. So what we have marrying these pieces of information is one, that there is this opportunity, but also that there is also a particular time point, which is like an on switch, where perhaps the benefit of any treatments will be particularly relevant. So what all this has amounted to is that preclinical Alzheimer's disease has been defined by the presence of the amyloid protein, and very much practically all studies nowadays require a level of confirmation of amyloid status in order for the compound to be considered to be treating Alzheimer's disease. And that goes back to earlier studies which show that if we just define Alzheimer's disease on a syndrome level, as we do in the clinic nowadays, actually only up to only about 30 to 40% of these people, when they go for amyloid scanning or when they pass away, if they go for autopsy, about a third of these people actually don't have the amyloid pathology, which puts a serious dent into the attempts of any compound that seeks to reverse this pathology to demonstrate action. So, as I said, so amyloid, being able to demonstrate amyloid positivity, as people have said before, either through CSF or PET, has become the mainstay of trials. And naturally, thinking about preclinical Alzheimer's disease, the next logical question is, what is the amyloid positivity rate among people who are in the preclinical stages? And this is one of the key meta-analyses from 2015, which showed that in a sample of, say, people in their mid-60s, there's a wide range of amyloid positivity, largely governed by this ApoE4 allele. But critically, if you don't have the ApoE4 allele available, only about 20% of people, which is by far the largest group of people, will be amyloid positive, which really reveals a major challenge in trials that try to run in preclinical Alzheimer's disease, because they need these cognitively healthy people to be amyloid positive in order to know that they're actually treating the underlying condition. So how to identify these people? We need scalable methods, because if everybody who we want to include into trial has to go through either a lumbar puncture or a PET scan in order to determine whether they're in preclinical Alzheimer's disease stage, clearly has limitations. So it's very costly, it's invasive, it's centered on a few centers in the country that can do these sort of tests. It's dependent, obviously, on highly motivated individuals. It also involves logistics, and it may well be that these people who are interested are actually the people who started to show symptoms. So maybe that they and that's what we're finding in studies, that they usually are in the advanced stages, which defeats the purpose of trying to run this study in the 10 to 15 year before onset of dementia range. Here comes, I would argue, digital technology through its cheapness, its ability to gather unobtrusive data. It allows remote testing where people don't have to come into into clinics, so they don't have to be within traveling distance of centers of excellence that can currently run these studies. It allows, importantly, I think, for societal reach. This is something we haven't talked about a lot yet, but I think it's very important because research, in-person research, remains the prerogative of affluent classes, and it really isn't very diverse in terms of ethnicity, which poses the questions about generalizability. And digital technologies, I think, here provide a novel opportunity to democratize research into Alzheimer's disease. It also allows testing into one's own environment, which is very important because for people who are in the preclinical stages of disease, it becomes very important. You know, the factors around the testing. Is there a white coat syndrome where they come in and they're being tested for hours in a clinic setting, or are they doing something on their mobile device in their own environment? And all of this opens up the possibility that actually these technologies may be particularly sensitive to the earliest changes, which then in turn may point us to these individuals and actually offer the invasive and expensive tests to those in which we're fairly certain that actually there is a bit of change in the cognition, which is very early, and maybe they're essentially if not preclinical, then at least in the prodromal stages of the disease as we currently define it. So there's a couple of different ways to approach it. The most obvious one is what can be sort of termed active cognitive monitoring, which is essentially, in its crudest form, it means transforming or essentially porting cognitive tests which are used in clinic, porting them into mobile devices, and then allowing people to do them remotely from their home. A perhaps more sophisticated way to do it is, for example, here, this is some work that we did where we actually thought, well, what are the advantages that the mobile technology offers that you cannot get with regular testing? And we arrived at the answer that actually using, for example, smartphone cognitive testing, you're allowed to test memory in, for example, memory or other cognitive functions at time scales which are simply unfeasible in clinical settings. So, for example, you could test the learning of you could learn some information and then test the recall of that information going in over the next 3, 4, 5, 6, 7, 8, 9, 10 days later, which, as you can see, is completely unfeasible if we were to do it in clinic. These are, for example, the tasks that we developed. One of them is where people essentially get to see some stories in a cartoon, and then they have to narrate what they see. So we're recording their speech, and then that allows us to analyze their speech later. And also it allows us to also test memory, because we ask them to narrate, to remember the story as they saw it. Then you have the gallery game, which essentially is a paired associative learning task. And then there's also an executive function task where essentially it's a version of the trail making task. So checking executive function, but using the ability of the phone to detect motion. And what we were able to show is that in a healthy group of people, so this is using the PREVENT study that Craig was talking about, we're starting to see a difference, which, for example, at baseline, people were pretty much performing at ceiling, but once you're able to use this mobile technology to detect retention in the periods up to 11 days, you really start to see a change where people gradually forget more of the information that they learn. And this has been acknowledged in the literature as accelerated memory extinction. So we all have, if you like, a point at which we've forgotten 50% of what we've learned. And this time point shifts earlier as we lose cognitive ability. So suddenly we have, using digital technology, this method of detecting very subtle cognitive changes, so changes to this 50%, if you like, half-life of your memory, that isn't accessible using traditional means. And then, this app that we developed was then piloted in a project with Alzheimer's Society. We had a very good take-up, so people really are very keen to help out with dementia research. And the thing that I want to draw your attention to is this heat map. So this is where in the country this app got downloaded. And that shows you that even though it's sort of focused on the main cities, it does show you also that it gets taken up in rural areas. And these are the people who you'd really struggle to reach otherwise for dementia research using traditional methods. Others have gone further, and that's one of the best validated apps, which relies on if you like, a form of VR, where you're using your device, which then projects certain objects, in this case a bear, and then using your camera, it instructs you to hide it somewhere. And then after a period, essentially the task asks you again, using your camera, to walk to the place where you saw you put this object, and then that gives you a very large amount of data in terms of your error, your spatial error, but also, for example, your gait and other variables. And these authors have been able to show that using this mobile app, they're able to distinguish with high degree of certainty people who go on to develop Alzheimer's disease versus those who don't. So this is, if you like, the active cognitive option. There's also passive cognitive monitoring. In here, the thing that's making the biggest strides at the moment is speech analysis, where people are analyzing the spontaneous speech of individuals using machine learning. They're able to extract certain features which then have been shown to be discriminative between Alzheimer's disease and patients. So you can easily see how this translates to the earlier onset, or to the earlier patient, the earlier stage of the disease, so preclinical and prodromal Alzheimer's disease, where again data is being collected, and we're again looking for patterns of change in one's voice as an early marker of cognitive decline. Other ways of acquiring passive cognitive information is also being developed. So modified browsers for PCs, where you can record the speed of one's typing, the speed of one's reading of articles and information. Also, the same things are developed for smartphones, where, for example, you could look at the time it takes to find names, or making spelling mistakes on testing. Other apps allow GPS tracking to give a sense of outdoor navigation, and as well as the time to get to a place that you're looking to find, as well as your accuracy in finding. And finally, another line of work is going around big data, where you're trying to associate, for example, internet searches related to cognitive decline to potential being a marker of potential early cognitive decline. Further examples of passive monitoring is something that we've been developing at Oxford, which is using wearable devices. These wearable devices are looped in with Bluetooth beacons that are spread around the house, so in specific rooms, so that effectively what happens is that when people move around the house, their signal is the signal from the Bluetooth beacon is being recorded on the smartphones, and then you can use this to triangulate the movement. So on the right, you've got the map of somebody who is moving around one of the museums in Oxford, and it shows you how you can create the pattern of movement, and then again it allows you to look at navigation, speed of movement, and look for markers of degeneration in that way. Then all of this really, so we're starting to get these very effective ways of collecting data at scale. So the next question is, how can we make this? How can we use this to accelerate research? And essentially, there's three models. One is where you get the people's consent on a, if you like, from the general public, and a project called Joint Dementia Research, funded by the NIHR, functions on this level. And essentially, in this case, you get some basic demographic and clinical data, as well as the consent of the person to be contacted about research. But because of its purpose to conduct things at a very large scale, you get very limited data. The third option is something that Craig talked about, which is an example is EPAT, where you Instead, you bring in people for extensive phenotyping as well as you genotype people who are at risk. You follow them up over a period so you can see what's happening to these biomarkers. You have an explicit consent to recontact them for these preclinical Alzheimer's disease studies. You have done some counseling around the genetic risk and amyloid positivity. And these are people who are, if you like, waiting to be contacted, and for whom there's a rich data that can be used to make the trials a lot more efficient, knowing each individual's trajectory. Now, the downside here is that, of course, these are very expensive projects that allow only a limited number of people to be recruited. And so we come to the middle option, which is what we've been working on, in which we reach out to cohorts which have gathered rich amount of data as part of their work, and we offer these participants to become part of a dementia-focused research register where we use their genotypic information, we use their retrospective information, but we also do some prospective data collection to allow very targeted recruitment studies. And this is where these remote assessments that I spoke about, so the active monitoring, the passive monitoring of cognition come into their own, because in a research registry of this size, so we're talking about 4,000 people at the moment, going on 5,000, you can deploy these remote technologies and start to harness data longitudinally, so that at low cost you can optimize your data set and you can recruit people into trials. This is an example of the flow of people. I won't take you through all of this, but the core principle here is that people get approached and then they give you very explicit consent to be recontacted on the basis of their data, specifically on the basis of their perceived dementia risk. And what we do is that we gather some data, and we also ask them to come back and to complete remote cognitive assessments regularly. This is what the database looks like today, where you have all of these different questions, all of these different data that you have on people. And then, for example, you can select people who have perhaps a level of cognitive impairment, which is subclinical. They're over the age of 60. That gives you the people that meet these criteria. You can see where in the country those people are, so that you can only focus on the people that would be within reach of your center. And then you get a lot of information on the age distribution. As you can see, we've been very lucky in attracting, if you like, just the right age group for the sort of studies that we want to run in order to accelerate dementia research. So people in their mid-60s, people in their 70s who are as yet cognitively healthy. And people here also in this solution allow us to ask questions about what types of studies they would be interested in, because really there is no point approaching somebody for a very invasive study if they've already said that they're not willing, for example, to consider a clinical trial or a positron emission tomography. So where does this leave us? Where are the interdisciplinary opportunities? One is that obviously the active cognitive monitoring scene is developing rapidly. We're moving away from companies being quite speculative in terms of the types of apps that they produce, and instead we're seeing collaboration between academia and software developers, where the claims that the apps make are essentially rooted into good science, and also the data that we get from it is a lot more meaningful. There's also opportunity around data complexity reduction using machine learning method. That virtual reality task that I showed you yields essentially more than 700 individual variables that need to be reduced in complexity. The passive cognitive monitoring, again, very exciting, but still the wearables need further work in order for us to extract reliable metrics of activity, sleep, navigational ability, and to link this to preclinical Alzheimer's disease, risk. Data mining is going to be very substantial over the next years through looking at this digital technology interaction that I was talking about, the speed of and way of interacting with digital technology as an early marker. as well as speech as an example. Then we need to do further work on the risk stratification algorithms. After all of these variables have to fit into some sort of prediction model, and that's where, for example, an interaction with the plasma biomarkers that Henrik was talking about, I think is going to be very influential, and there's some early work that these plasma biomarkers improve these algorithms. And we can also think about going beyond ApoE4 and thinking about polygenic risk scores. And then finally, a further opportunity is to focus on research infrastructure, working on effective and secure data linkage, specifically around linking the data, the research data of the volunteers that have graciously agreed to help us to the vast data that's held on them through healthcare records. And that's what I wanted to talk to you about. I'm very happy to take questions. I hope that was clear.
Thank you very much indeed. That was an eye-opener. I can remember going to a meeting with Paul Matthews. It must have been about a decade ago now, and listening to all the excitement. And Paul was very excited about e-health and remote monitoring opportunities. And your talk really shows that this has really taken off, and a lot of the excitement has now been realized. So that was extremely impressive. And I guess from the point of view of the physicists, you alluded to this, you have the opportunity to provide very densely sampled time series, which means one could, in principle, start to identify a very trajectories at a very fine-grained level. So hopefully you will also be available at 3 o'clock just to sort of review some of these issues. And let's now move to Manchester, and it's my great pleasure to introduce Laura Parks. Just pursuing the interdisciplinary theme, I can't resist noting that Laura has masters in both physics and neuroscience, and is now neuroimaging neuroscience lead in Manchester. And what she's going to be talking about or focusing on specifically is the role of microvascular dysfunction in neurodegeneration. Laura, when you're ready, we can see your slides. You're ready to go.
Thank you very much, and thank you very much for the invitation to speak today. So I'm going to talk today about the use of MRI to measure vascular dysfunction, which is something we haven't talked about so much today. In the clinic, clearly use MRI to look at vascular lesions in the brain, as shown here. But we can also look at measurements of physiology from the normal appearing tissue. So, for example, an image here of cerebral blood flow. But MRI, there's continual innovation in MRI, which is one of the points I want to get across. And I want to describe today some of the ways we do this and the new measurements of vascular dysfunction that we're developing in Manchester. So I lead a team of physicists and biologists, and how we go about developing new measures, we come up with a biophysical model. So for example, here of water movement in the microvessel network. We can sensitize our MR measures to different properties of this model. And then we implement this on the preclinical system so that we can validate the measures in animal models, which I think is critical for MR, where the signal really could come from many different sources. So validation is really important. And then hopefully at the end of this process, we have quantitative, accurate, sensitive measures of brain physiology. So how important is vascular dysfunction in neurodegeneration? Do we need these measures? I think there's really a wealth of evidence now for the role of vasculature as a contributory factor in all dementias. So it's nicely summarized by a review a couple of years ago, where actually the JAK model that you may all be familiar with was updated to suggest the inclusion of an early vascular component. So this may really be a factor that changes early in the disease process. How does vascular dysfunction affect cognitive impairment? Well, this is a lot of unknowns here. This is potential mechanisms that have been put forward in small vessel disease. And I think there's a lot we don't know, but I'm showing this partly because there's a lot of disease processes here that we can access with non-invasive imaging. So with MRI and with PET. And so through use of, for example, simultaneous PET-MR, we can more fully characterize disease pathology in the brain. This would allow us to study biological processes in the human brain, but also potentially produce biomarkers that could be used to test new therapeutics that might impact on some of these mechanisms. Ultimately, of course, also could characterize disease fully in clinical population or individual people. So I'm going to focus now and just describe some of the physics innovation and the new measures that we've been developing. And then I've got a proof of principle study where we've applied these in a small group of people with mild cognitive impairment to test their sensitivity. So first of all, measures of blood-brain barrier dysfunction. So the standard ways to measure this using gadolinium contrast agents. So you can collect a dynamic series of brain images and inject gadolinium. And you can see here the signal in the tissue increases as the contrast leaks out of the blood-brain barrier. And here's an example of a map. You can produce a quantitative map of that leakage shown here in stroke. And this is the signal time course you might expect compared to the situation of no leakage. But the problem comes when we look at this in mild cognitive impairment and in healthy aging, where it becomes increasingly difficult to detect these very low levels of leakage. And you need to scan for a very long time and have very stable measurements in order to detect this. So we wondered whether there might be a way of using MRI to provide a more sensitive measure of blood-brain barrier dysfunction. And this is work led by Ben Dickey and also in collaboration with Jeff Parker at UCL, where we've looked at ways to try and sensitize our MRI measurement to water exchange. So rather than looking at leakage of gadolinium molecules across the blood-brain barrier, which is a very slow process, instead we think that looking at water exchange, which has many more routes to cross the blood-brain barrier, may provide us with a more sensitive measure and a faster measure of these very small dysfunction in the blood-brain barrier. And there's different ways we can manipulate our MRI experiment to sensitize us to water exchange. So we need to isolate somewhat the water signal from inside and outside the vessel. So one way is to use arterial spin labeling to isolate the signal in the vessel. Other approaches can isolate the water outside the vessels, or we can look at ways to accentuate the difference in properties, T1, T2, diffusion between the water in the two compartments. So Ben implemented one of these techniques on our preclinical system, and we looked to see whether it was sensitive to blood-brain barrier dysfunction in an Alzheimer's disease model, a rat model. And shown on the left here is the permeability surface area of water. And you can see indeed it is elevated in the transgenic rats compared to the wild type littermates in white. And comparing this to standard gadolinium leakage, we weren't able to detect differences using that technique. So it is suggesting that these water exchange measures are more sensitive to these subtle levels of degeneration that we may see in dementia. Furthermore, we were able to validate the measure against immunohistochemistry measures of tight junction protein occludin. So moving on from blood-brain barrier dysfunction to look at further measurements we can make of the microvascular network. Arterial labeling is a way to measure cerebral blood flow. And this involves creating a labeled bolus of blood water, and then you look at how it changes the signal in the image slice. But if you perform this experiment with multiple delay times, as well as measuring blood flow, it allows you to also measure this arterial transit time, which is the time that it takes blood to reach the capillary bed. And it's weighted really towards the smaller vessels as the time is longer down to those small levels. And you can see here a typical map of arterial transit time, showing increased transit time to some of the watershed areas in the brain. And in some other work in Parkinson's disease, we found that we see quite pronounced prolongation of arrival time and only very small changes in blood flow. So it suggests to us that transit time may actually be a more sensitive marker of vascular dysfunction than blood flow. And then another technique looking at microvessel structure, so this idea that transit time is prolonged, got us thinking about whether it may be due to increased tortuosity of vessels. So Lauren Scott, a PhD student in Manchester, has been looking at changing the diffusion weighted imaging protocol to try and access signal from the perfusing water. And by changing the diffusion time, so the diffusion gradient sensitize the measure to movement of water. And if you can change the diffusion time, you can sensitize to different amounts of movement. So for example, at short diffusion time here, the water will just be moving in one direction. As you increase the diffusion time, you're going to get changes in direction of those water molecules. And you can model this, you can use physics models to model this process. And if you collect imaging measurements at a range of diffusion times, you can then get access to features of this network. So for example, the perfusing fraction, so the amount of water in blood, and also the capillary segment length. And again, Lauren's looked at validating this in, on the preclinical system. So we used, again, rat brain, and this time we were able to create vascular casts of the brain. So there's a photograph shown here, and then a very high resolution micro CT image of that brain, where we can really see right down to the capillary bed and get a ground truth estimate of the capillary segment length in different regions of the brain. And we were able to compare that to our MRI measures shown in this graph here. And you can see across 4 different brain regions, quite nice correspondence between the ground truth CT and the MR measures. So we're really able to infer quite microscopic properties of the tissue from the MR signal. So final measurement we've developed is looking at brain oxygenation, which is also potentially very important. And John McFadden, PhD student working with me, has been looking at the use of quantitative susceptibility mapping to access measures of oxygenation. So this shows you a susceptibility map of the brain. You can see very high signal in the veins in the brain. And this is because of the presence of deoxyhemoglobin. And so we can quantify in the vessels the concentration of deoxyhemoglobin, and from that get an accurate estimate of oxygen extraction fraction. And John's been looking at how accurate this is for different sizes of vessels to see if we might actually be able to get more regional information from smaller veins. So moving on now to just describe an experiment we've just completed looking at sensitivity and repeatability of these measurements. So we've implemented these measurements on simultaneous PET-MR scanner in Manchester, and we've studied, it's a small study, we've studied 13 people with mild cognitive impairment and 16 age-matched controls. We've characterized the vascular risk, cognitive tests. For PET, we've used flutometamol injection in order to get information on amyloid status. And then the MR included all of the measurements I've described and more standard imaging measurements. The key questions are, how repeatable are these measurements? How sensitive are they to vascular risk? And how sensitive are they to cognitive impairment? So just present the results here. So first of all, looking at the top line here. So these are all our measures of vascular disease. And this shows, first of all, the repeatability measures, so the coefficient of variation of these measurements. Because for a technique to be useful, we really need decent repeatability, or we're just measuring noise, really. And I think it's quite telling that the gadolinium leakage measurements were really quite poorly repeatable. Again, I think because we're looking at a group of people where there's very little leakage of gadolinium, and we're just not able to detect that in a repeatable way. Whereas our water exchange measurements are still not brilliant, they're not fantastic repeatability, but they're better. Some of the other measurements have got quite reasonable repeatability. So moving on then to look at the relationship between these measurements and vascular risk. As expected, a number of the measurements show a relationship with vascular risk. And I've pulled out the significant ones here at the bottom. So you can see increased white matter lesion volume with vascular risk, in prolonged transit time, as we expected, increasing perfusing fraction, so it's suggesting that there's actually an increase in blood volume with vascular risk, and increased oxygen extraction fraction. And then finally, looking at the sensitivity of these measures to MCI. So we looked at the difference between the MCI and the control group. And again, a number of the measurements are sensitive to this difference. One thing to highlight is, while the water exchange time had quite poor repeatability, it showed really quite a large change between the groups, so over 100% reduction in exchange time in the MCI group. So water is crossing more quickly due to blood-brain barrier dysfunction. Other thing to notice is that the most significant measure, again, is this prolongation of transit time. And we wanted to look at what might underlie this. So look at the relationship between this measurement and some of the other vascular measurements we've made. And one thing that really stood out was the relationship to perfusing fraction. So you can see this here. So people with longest transit time also have the highest perfusing fraction. So it suggests that transit time might be prolonged because of some chronic vasodilation, which is slowing down the movement of blood. We also found a strong relationship between transit time and oxygen extraction fraction. So it's possible that this longer transit time in the capillary bed is somehow aiding the extraction of oxygen. And finally, one of the things struck me as being quite interesting was the fact that oxygen extraction fraction was highly related to vascular risk. It didn't seem to confer, it didn't show any difference between the MCI and control groups. And I think this puzzle is solved, I think, if you look at the relationship between oxygen extraction fraction and blood flow. So you can see a negative relationship. So people have reduced blood flow, they have compensatory increased oxygen extraction fraction. And this results in a fairly stable level of oxygen metabolism. So why this isn't really contributing to cognitive impairment. So finally, we looked at putting the independent vascular imaging measurements along with amyloid status into a multivariate model to see if we could predict the MOCA score as a dependent variable. And just using these four measures, so amyloid, transit time, white matter lesion volume, and water exchange time, we're able to predict 57% of variance in the MOCA score. And it's showing really that these vascular factors are having a larger effect on cognitive impairment than amyloid status. So just to wrap up, I think while this is a small study, I think we have shown evidence for vascular dysfunction in mild cognitive impairment, including both blood-brain barrier breakdown and also hemodynamic changes. And MRI can provide sensitive measures that characterize a range of these vascular properties. One striking thing to me was the prolongation of the transit time in mild cognitive impairment and how it's closely related to chronic vasodilation. CBF is reduced as expected, as has been shown in a number of previous studies, but this is compensated by an elevation in oxygen extraction fraction, so maintaining baseline oxygen metabolism. So one question is, if all these vascular changes are acting to preserve baseline metabolism, why are they then related to cognitive impairment? Well, I think what might be happening is that this chronic vasodilation is leaving very little capacity for the vascular system to respond to neural challenge. So again, studies of cerebrovascular reactivity have shown this. So it might be interesting to look at, for example, whether prolonged transit time is related to cerebrovascular reactivity. I think, as a number of other people have said, we need to move to longitudinal studies to really look at the interaction between these factors and look at time course of these changes to really understand the impact not just on cognitive impairment, but on cognitive decline. So thanks for listening, and just like to acknowledge a whole host of people who've been involved in this work. Thanks.
Well, thank you. That was marvelous. A beautiful demonstration of how physics can really inform the interpretation of data. If I read you correctly, you've been able to reconstruct the wrinkliness of the vasculature, which I'd never heard before. That's brilliant. You, I hope, will be available at 3 o'clock just to sort of drill down on some of these things. So thank you again. Just to sort of survey what we've enjoyed in this session, I think we've had a really nice multilateral perspective that emphasizes the interdisciplinary aspects of why we're here. We've had sort of data science from John. We've had chemistry from Enrique. We've had digital opportunities from Ivan, and I repeat, a beautiful demonstration of how physics can really sort of add up our game in this joint endeavor. So thank you to all the speakers of this session. That was brilliant. We're now going to take a break for lunch. We're going to reconvene at 2 o'clock, and then my colleague John O'Brien will be taking over the chair, and then we'll review all of this session's talks and our speaker panel questions and answers at 3 o'clock. So thank you. See you in a few minutes' time. Bye-bye.
Hello, everyone. I'm John O'Brien. I'm Professor of Old Age Psychiatry at the University of Cambridge, and it's my pleasure to welcome you back to this afternoon session. I'm sure you'll all agree we've had a fantastic morning, and I know we're going to extend that to the afternoon. We've got 3 speakers followed by a question and answer session. So please do continue to send your questions through the Q&A, and then Carl and I will address those with the panel all together between 3 and 3:30. So it's a pleasure to welcome our first speaker, Zoe Courtsey, who's a colleague of mine here at Cambridge. She's Professor of Experimental Psychology in the Department of Psychology, and her research interests are in bringing together cognitive and behavioural data together with imaging and others using computational neuroscience to look at learning and plasticity across the lifespan, with a particular interest in later life and cognition, which is the topic of her talk today. So Zoe, welcome, and we look forward to your talk on machine learning for predictive prognostic trajectories in dementia.
So I just wanted to start by summarizing some of the key challenges that we heard about this morning when we try to put together data that come from many different modalities, markers that come all the way from imaging and genetics and even digital data, and trying to extract sensitive features that allow us through modeling to predict early progression in neurodegenerative disorders. So some key challenges that we encounter is first that we deal with heterogeneous populations, primarily due to comorbidities. Second, that most of the clinical data that we have are sparse and incomplete. Some of the scales based on which we measure cognition are not very sensitive. And at the opposite, some of the biomarkers are measured with quite costly and highly invasive tools. So moving forward, what we really need is solutions that allow us to do early precision diagnosis and patient stratification based on as least costly and least invasive data, with the hope that this will guide clinical trials to be more cost effective and ultimately personalized interventions. And what I'd like to do today is give you some examples of how we can use machine learning to derive this type of solutions, and to derive solutions that are transparent, they are clinically relevant, they are interpretable, and they're also interoperable. So focusing on the problem of dementia, I think the key challenge is actually nicely described in the Jack curve. What we see here is that our challenge is to predict and diagnose as early as possible. However, the markers that would allow this are biological markers that are highly costly in their measurements and highly invasive. And as we move forward in the progression of the disease, we have MRI data and we have cognitive testing data that are less invasive and less costly, but with raw eyes are less predictive. So how can we solve this problem of early prediction while imposing as least invasive tests to our patients and saving costs for the NHS? Another challenge is the problem of misdiagnosis, which in machine learning is translated to misclassification. So here I illustrated an example of a discrimination of classes of patients at early stages of neurodegenerative disease. We're looking at mild cognitive impairment, and we want to be able to separate patients that will remain stable, while in contrast to patients that might progress. And this is a very difficult problem. Currently, we try to solve this by using clinical labels and defining some rather arbitrary cutoffs. So for example, if a patient carries the diagnostic label of MCI for more than 3 years, then we consider this patient to be progressive, MCI progressive, while if a patient during this period has remained stable, we label this patient as a stable MCI. However, this kind of toy diagram here shows that a patient around the boundary of this type of classification, who's labeled as progressive, might be very similar in their profile as a stable MCI patient, and might be quite different from other patients diagnosed as progressive MCI. This causes a lot of problem in terms of misdiagnosis. Comorbidities here play a major role, and our drive with building models that allow early diagnosis and prediction is really to try and reduce this misclassification risk. So how can we do this? There are many different approaches, and it's very exciting to see the field moving forward in building models that are highly predictive. I'd like here to show you an example from work that we've been doing with my team. Our work focuses on a framework that involves 3 phases. In the first phase, we extract features that are highly predictive of changes in cognition. And in the second stage, we use a machine learning algorithm that allows us to make reliable and robust predictions. And in the third stage, we move away from binary classifications to a trajectory modeling approach that allows us to reduce the risk of misclassification. So in the next few slides, I'd like to take you through the steps of this approach. So as I said, a first step is to extract features that are highly predictive. We focus primarily on structural imaging data. Structural imaging data is easy to acquire, less invasive than, for example, PET. It takes about 5 minutes to acquire a good structural scan, and therefore can provide us a least invasive and costly marker. So what we do here is we apply partially square regression. We used for building our models the ADNI dataset, and we use about 500 individuals. We use their baseline MRI scan, so as early as possible, and with applying partially square regression on the structural scans of these patients, we derive a gray matter density score, which is predictive of changes in memory for these patients. Now, what this allows us to do is when we use that in a large number of individuals, a large cohort, then any new patient or any new individual that comes in, if we have a T1 scan, we can extract this predictive gray matter score for this patient by comparing their data to the model data. This score that we get for gray matter density, which is derived based on the PLS regression, we saw here that it predicts cognitive decline. It accounts for variance in the ADNI-MEM scores. And this is within sample validation. Also here I show you out of sample validation, we take a different data set from ADNI, and you can see again here the relationship between the PLS-derived gray matter score and the ADNI memory scores, and also using a completely different data set from Nimrod, thanks to John. We see here again with a different actually scale with ACE-R. Here we see that for new patients that we did not use to build our gray matter feature, we can also predict changes in cognitive performance for these new patients based on their T1 scan. Now, once we know that we have a reliable non-invasive feature that allows us to predict changes in cognition, we bring this feature together with other data that I'll show you in a minute into a machine learning framework. Here we used a rather shallow model that is easily interpretable, and effectively what we do is we build two prototypes, one per class. If our classes are, for example, stable versus progressive MCI, we have two prototypes, and any new training data that comes into the model allows the prototypes to move apart and determine distances between these two classes. So how do we build our models based on this framework? We built what we call a biological model. This is a model where the predictors are primarily biomarkers. In this model, we put our PLS-derived gray matter score, we put beta amyloid, and we put APOE4. And what you see here is we test the model on a rather fine classification. We tested in separating two classes, stable from progressive MCI. And you see that the model does well on this. It predicts with sensitivity and specificity higher than 80%. This separation between these two classes. And the nice thing is that you can interrogate the model, and you can see based on the tensors the contribution of each one of these predictors to this classification. And for example, you can see clearly that beta amyloid is got a strong weight of contribution into this prediction. Now, as I said, we are very much interested in building models that use less invasive and less costly markers, and therefore we ask whether this biological model is absolutely necessary, or whether we can do the same thing with cognitive data. And what you see here, again, working with the ADNI data, we built a separate model using exactly the same framework, but now we include only cognitive data. We include the ADNI-MEM score, we include executive function scores, and geriatric depression scales. And you see that this model, actually the cognitive model, does equally well in this classification of progressive versus stable MCI in terms of sensitivity and its specificity. Again, we can interrogate the model and see, for example, that the memory performance, the memory scores are key for predicting classification. So up to now, what I've shown you is an approach that takes different markers and makes a binary classification. But this type of binary classifications, as I hinted earlier, carry a rather high risk for misclassification. For example, we know that a classification of MCI has about 30% potential misdiagnosis. So how can we move away and move away from these binary classifications based on clinical labels and reduce the risk of misdiagnosis and misclassification? We've taken a trajectory modeling approach, and we derive a prediction score based on all of these multimodal data that are integrated by our model. And I will just simply describe it here. Effectively, what we do is our model creates a prototype for stable health. What would an individual look like if they were a stable patient? And with a trajectory modeling approach, we derive distances from this prototype. Every new individual that comes in, we put their data in the model and we measure how far they are from this stable cognition prototype. And we don't only measure the distance, but we also measure how quickly they move away. So we measure a rate of change, a rate of decline. And this trajectory score allows us to make the diagnosis in a more personalized way for each individual, moving away just from classes and labels. And what you see here is we can derive this trajectory score based on the cognitive model I showed you, or based on the biological model. The cognitive model just has cognitive data. The biological model has different types of biomarkers. And what you see is that the further away you're moving from the stable prototype, the higher the rate of memory change, the higher the rate of decline in cognition. And this is similar for the cognitive model and the biological model. However, what we see is in the biological model, we actually have a stronger relationship, suggesting that for trajectory modeling approaches, biological data, biomarkers can be more sensitive than cognitive data. While when we just make a bit coarser class-based discriminations, these two models seem to be quite similar. So how do we validate this trajectory modeling approach? What you see here is we take data from a separate group of ADNI again, and we don't train the model on this data. We simply test. This is a kind of new data where we put it in the model and ask the model to build this relationship. And what you see here is that this projection approach creates a score, a trajectory score that accounts for 46% of the variance in rate of memory decline. And we also compare this trajectory score to changes in other biomarkers. For example, we see a correlation with beta amyloid. The further away you move from the stable prototype, your score will be higher and correlates with beta amyloid. It correlates with atrophy in medial temporal cortex. And also we see when we look at groups of individuals that they split, that they're split based on APOE4 positivity, we see that the trajectory scores are very different for these individuals. So this really validates that our trajectory modeling approach relates to dementia pathology. More interestingly, given that we want to move as early as possible, even at pre-symptomatic stages of the disease, what we did here is we used the trajectory score in the ADNI data to classify patients into different groups. Patients that have already diagnosed of AED, early AED, and stable cognition patients. And you can see as the scores, how the scores distribute. But also we look at healthy individuals and mild cognitive impairment individuals in ADNI-3. And in a Berkeley cohort that comprises of only cognitive normal individuals. And you can see that we can split individuals into different classes based on the trajectory score, even at pre-symptomatic stages. So the next step that we were interested in doing is in predicting not only cognitive decline based on cognitive scores, but also on a biomarker. So for example, FTG tau. And we follow the same approach. The model is built as I showed you. We don't train the model anymore. The model is trained. But now we test the model with ADNI-3 data we did not use for the training. And we use our trajectory modeling approach to actually test for changes in tau. And what we see here is that the multimodal trajectory score that we built through our model predicts regional changes in tau over time for individuals that are at early stages of AD. And you see here, for example, correlations of this trajectory score with tau in the temporal cortex and the parietal cortex. And what we see is that individuals at early AD stages accumulate tau 2.8 times faster than individuals that show stable cognition. We then move to do the same with a Berkeley cohort that comprises only of cognitive normal individuals, and we see a similar picture here with a trajectory score predicting the rate of future tau accumulation in these individuals that are at the pre-symptomatic stage of the disease. And finally, we test the utility of our model, particularly in the context of potential use in clinical trials. And what we see here is looking at regional tau. On average, we see 35% reduction in sample size when we measure changes in tau versus cognition based on gold standard PAC scores. But also we see a mean 30% reduction in sample size needed in a clinical trial to measure changes in tau when using our model trajectory score compared, for example, a biomarker, for example, a beta amyloid. So just to summarize, I hope I've given you an idea of how modeling approaches can help us define prognostic trajectories by combining and harnessing the power of multimodal data, reduce the risk of misclassification, improve patient stratification, generalize predictions as early as pre-symptomatic stages, and might also have potential for the sensitivity of clinical trials, and also reduce costs in healthcare. And our vision really for the future is to be able to use these approaches to build clinical decision support systems that are AI enabled. They demonstrate transparency and reproducibility. They help clinicians to assign the right patients at the right time to the right diagnostic and treatment pathway, improve patients' well-being by allowing patients to participate in less invasive and less expensive diagnostic tests, and potentially guide more precise selection for clinical trials. So I'd like to close by thanking all our collaborators, clinical colleagues, and mathematicians, computer scientists. My lab's been working under pandemic conditions to deliver all of this work, and all our funders for obviously supporting the work. Thank you for your attention.
Thanks very much, Zoe, for that fascinating talk. It's great to see how you've brought together cognitive behavioral imaging and other biomarker data to show how it can be applied to clinically important problems and ultimately aim to improve patient care, which is obviously what we want. So thank you very much for that. Also highlighting some of the gaps, including some gaps in clinical data, which I hope if we've got time, we'll be able to come back to in the panel discussion. So thank you. I'd like to now introduce our next speaker, who is Dr. Viola Priesteman. Viola is head of the Theory of Neural Systems Research Group at the Max Planck Institute for Dynamics and Self-Organization in Göttingen. Her group there works using methods such as statistical physics and information theory to better understand how neuronal systems are organized and how they can be optimized for neural computation. And her talk today is on what statistical physics can tell us about living neuronal networks. Welcome, Viola, and we look forward to your talk.
Yeah, thank you very much for the kind introduction. I think I'm here a bit the oddball in this entire talk, but this entire conference, but I'm trying to give you an introduction of how we approach from statistical physics fundamental aspects of brain function, and I hope that afterwards you understand on the one hand our working, on the other hand a bit about the basic dynamics that we find in neural networks. This title of the talk has been basically almost given by the organizers, and I thought I might try to go in that direction, say, what can statistical physics tell us about living neural networks? My group basically uses statistical physics, so this is my statistical physics of neural systems, but it's really also very typical approaches from the field. So just to give you an overview about what are the typical topics. So phase transition play a role. Phase transition is whenever in any media something fundamental changes. For example, a system is quiescent and then becomes really responsive. There is no specific patterns, but then patterns start to emerge. These can be very abstract patterns, like how would one encode information in a network. This is, in an abstract way, a pattern, even if you don't see it with the naked eye. To basically be able to see this, we can make use of information theory to quantify, but also then to design computation. I would still put that in the wider sense into the statistical physics, because we're applying it on these large systems. Well, in principle, statistical physics deals with 10 to the power of 23 particles, so huge systems. That's the Avogadro number. In that respect, the brain is still very small, and what we can record from the brain is even less. So when assessing single neuron activity with extracellular electrodes or with calcium imaging, it's typically still only a tiny fraction of the entire brain that we can assess, apart from a few very, very small brains. And then what I already mentioned is pattern formation and collective dynamics. That's what a physicist might call learning and local learning rules. I would like to show you what the challenge is to bridge from statistical physics then to neuroscience. You might all know this beautiful calcium imaging from the zebrafish larvae. Here's the two eyes of the zebrafish larvae. It's the top view. It's a time-lapse video, and the colorful illustration shows you how neural activity very often is quite local, but sometimes also spreads over the entire system. When we assess this type of activity in this kind of a zebrafish larva, there are about 100,000 neurons and then thousands of connections per neuron. So that is already a highly connected system. The interactions are very much different from what in principle you would know from physics. Just to tell you where we start from when we try to apply these physics approaches. They are pulse-like. So normally if you think about about your physics classes, there's continuous interactions when two, or there's and they're directed. So let's say one neuron can activate another neuron, but there's not necessarily, as you know, a connection in the opposite direction. That makes it difficult in terms of using symmetry laws that we typically make use in physics. The interactions are time delayed because one neuron is active, and then it takes a few milliseconds until that spike has traveled to the subsequent soma. And then all this change over time. So there's not the one strength of interaction that stays there forever. Nonetheless, so overall, this makes these classical symmetries and conservation laws like energy conservation. That's nothing that we can apply to the propagation or to understand the propagation of activity, information processing in neural networks. Obviously, energy conservation holds in the brain, but it does not help us much in understanding the activity propagation, because that's an open system, there's energy supplied, and there's no reason why one spike should only lead to one other spike somewhere else. And then last but not least, obviously, we would love to follow every single spike in this high-dimensional network. We would love to know which neuron is connected to which. But because it's this high-dimensional topology, because any neuron could in principle be connected to any other neuron, and we can't access it, it's quite difficult, obviously, to characterize these collective properties. That's where we are starting. I mean, this is very different from statistical physics that you might know classically, where we treat gases and the statistics of these ones. So what I want to do today is to go through one very basic approach, where we take this reductionist approach and wonder, how does activity propagate in the network? And what can we then learn about abstract information processing in that system, but also about the stabilities of that system. So the first part, therefore, is to go to quantify the spreading dynamics, and then the relation between exogenous and endogenous activity. So take a brain area, many, especially in cortex, we have highly recurrent activity. Part of the activity thus is coming from outside, another part is really internally generated. That relation plays a role for information processing. We then analyze neural data from macaque monkey And we then go again to a model, because there we have better control overall, and then show that information transfer and also task performance depend on this relation between the input strength and the recurrent activity. So, propagating activity, why are we interested in it? Take cortex, take your favorite neural model, now assume that there is one extra spike, like the Mickey London paper from 10 years ago. There's one extra spike, and depending on the postsynaptic neurons, this one extra spike might activate other excitatory, or this one extra spike in the excitatory neuron might activate other neurons, and thereby some perturbation, some cascade of activity might run over the system. And potentially this cascade at some point dies out. And mathematically, we can describe this kind of a perturbation fairly well. And we can describe it very discreetly here within the framework of what's called branching process. And the dynamics are separated into 3 different groups. It's fairly clear that if you perturbate one neuron, and this leads to, in the next step, more than one neuron being active in the next time step, and these again activate more neurons, then activity would increase. Here, I don't explicitly speak about inhibitory neurons, so take this as effective activation. You could imagine that such a activation could become stronger and stronger, even with inhibition modulating that. So think about it of effective excitatory activity. So one neuron could in principle activate more than one, and this could in principle amplify. If one neuron on average activates less than one in the next time step, then such a perturbation would die out. And this 2 different phases, one where any perturbation would grow to some degree, obviously there are some compensatory effects at some point. Let's say initially it would increase, or the point where it would decrease. This is separated by a critical state, which you might have known of. And I want to take all the voodoo away from this criticality and just say, this is the state where the system is quite sensitive to input, but but is not yet unstable. It's at the brink of becoming unstable. In neuroscience, as you know, people are interested in that critical state because it has been associated with certain information processing properties. We'll come to that later. So now, if we would like to know whether this plays any role, if we would like to know whether knowing about that propagation does tell us anything about cortex, it would be great to know in first approximation what this branching parameter is. Is it above 1, at least shortly? Is it below 1? How far is it below 1? And we developed an approach how one can estimate that, even if one only sees a tiny fraction of the network. If we would see the full network, it would be very easy to estimate it. You take the activity at the subsequent time step, the full network activity, and correlate it with activity at the previous time step. And then the slope here, the correlation between the full network activity, really summing over all neurons, that would give you specifically this branching parameter. If you from the entire network only see a tiny fraction, then this correlation looks much smaller. This is, on the one hand, a problem. So you basically here in this entire branch don't see all the spikes, but you only see a fraction of them. Then you see a smaller correlation, and if you would see the full network. And that means if already the correlation strength is biased, it's hard also to make inference about the full system. The good thing is we managed to overcome that subsampling bias and find a way of how we can very precisely estimate here this branching parameter that tells us how much activity propagates from just a tiny fraction of the entire system. So I will have to cut the math short here, and I think that's also not of the core interest. I want to give you the properties. So we can estimate this control parameter, and it is fairly efficient, it's fairly precise, and it's easy to apply. The only thing that one needs to know is the number of spikes at a certain time point from all the neurons recorded. So in principle, you don't need spike sorting. It's not required to know the system size. It's not required to know how many units you sampled or any moments of the process. All you need to know for every single time when you define is how many spikes did you see. And from that, you then infer what is the propagation of activity in that network. Under ideal condition, and with ideal, I mean really in a network where all, in a theoretical network where all neurons are like, in principle, a single neuron would be sufficient because you can estimate that from the return times. And this, we do have a toolbox out for anyone who's interested in the related paper that describes it step by step. So we can have an estimate under the assumption that there's propagating activity in the network. And we assume, I mean, I started by saying, well, there's a small perturbation by one external input. We just assume that comes in either from other brain areas or from the sensory periphery. So these perturbations are occurring anytime, and therefore it might be reasonable to approximate the system with such a branching framework. So that's the first step. We quantify spreading dynamics. And now the question is it useful? I mean, all models, we can make any model. And a beautiful saying says all models are wrong or all models are limited, but some are useful. How useful is this way of thinking about network activity? So my group, we applied that estimated to a number of different cortical recordings, to cortical recordings from rat, cat, monkey, and human. And we find that this branching parameter is around 0.98. So it's close to the one where one would be at the critical point. From theory, we also know that the information transfer in the network, it strongly peaks the closer one gets here to this transition of instability. So it seems that cortex in these different animals is not right where it would maximize the information transfer, but it's in a regime where it could, in principle, strongly tune the information transfer. It could be at high or at low information transfer. We go back to that. This already hints that there is no need to maximize a certain property like information transfer, but it would be sufficient to, for example, have moderate information transfer in that aspect. How would such activity look like if one is close to the transition? It would say that if you would have the ability to have the sum spiking activity of the entire brain area, it would fluctuate considerably. Here's the time, and here's really the total number of spikes that we see at every single time point. This is a model, and we would expect such fluctuations. I should have put the spectrum. This gives a 1/f power spectrum automatically. We don't expect that it would be fairly unordered, Poisson-like. We also don't expect that it would be near a critical point, which is also a popular hypothesis, because then we would expect long pauses between these bursts of activity. In vivo, there's definitely the continuous type of activity. Now where we know this number, 0.98 instead of 1, so 2% away from this critical transition. What can we do with it? First thing we should do is validate the model and see whether it's a good approximation to neural activity by looking at other properties. And one other property is, for example, the autocorrelation function. The autocorrelation function over time, in milliseconds, in the model decays exponentially. And this is exactly what we also find in spiking activity. But note here the y-axis. This autocorrelation function does not start at 1, it starts at lower values. That's something we also find in both. Then we could ask, like, how many units do we need to sample and to estimate here the decay constant of this exponential, for example. And here in that model, we can take 50 of the 10,000 neurons, 25 or 1, and we will always get these the correct and consistent intrinsic time scale here. And the experiment, because of diversity, you see how the variance gets larger and larger, but maybe down to 13 neurons would be sufficiently to have a consistent estimate there. The variance also gets larger because we decided not to take longer recordings, but only to take fewer neurons, so we basically have less data. Going to the autocorrelation, I mean, everyone who has looked at autocorrelation functions of spiking knows that typically this is how it looks like. There's a small, strong peak at 1, because, at 0 time lag for definition. But if you then zoom into the floor here, mind the floor, one finds this exponential decay in, let's say, more than half of the neurons. And this kind of exponential decay, this is what we expect from theory. We expect that in such a recurrent network, if it is considerably branching and propagating activity in the network, we expect here this floor, this strong decay of the autocorrelation function, and then this exponentially decaying floor. Sometimes, in addition, you have these oscillations on top of that. So it's expected to see this type of autocorrelation function when sampling only one or only a few neurons of a large network. And there's still a reliable exponential decay from which one can principally infer the spreading dynamics. So a model of spreading dynamics closely matches the number of spike statistics to name a few. So we have in blue always the experiment, in red we have the model where we think we have the best fit. Inter-spike interval distribution, here the blue and the red match well, but it also would match with alternatives like fairly irregular or near critical dynamics. The number of spikes per bin is well matched. The avalanche size distribution, I mean, from the field of criticality, is really well matched. The same here for the avalanche durations, it's well matched. And also the cross correlation between any pair of neurons here in blue and the red model and experiment are fairly well matched for any of these bin sizes. So these are all properties that we did not put in. We just said there's spreading dynamics. We want to know how much there is of spreading. Do we, or can we, I checked into that, sorry. Can we infer any properties of it? And you can see that the number of properties of basic spike statistics can be matched from just estimating how much a single neuron might perturb such a network. Now, ideally, we would like to predict properties that we don't know yet. One of the aspects we can predict are, for example, here the question of perturbation. We perturb the network, like Vicky London did it a few years ago, and then ask what would be the firing rate in the full network. And we would say, well, there would be a perturbation that will last for a couple of 100 milliseconds. You would see it. But to do that, it would be fairly tedious experiments. The other thing that we can now ask is, what is the relation between exogenous and endogenous activity, in the sense that there's input from other brain areas, or there's recurrent activation within the brain area. And we find from macaque prefrontal cortex, we would predict, it's model prediction, it would have to be tested experimentally, that between 1 and 10% makes the external activation. In hippocampus, it seems that it might be even less external activation, even more recurrent aspects. We can go further and ask, is it the same for all cortical brain areas? And here we have a unique data set from our collaborators, Charlie Gray in Montana and Nick Dodson. They recorded spiking activity throughout the entire cortical surface, and we find that the visual cortex is basically further away, has a bit less of internal recurrence, whereas the frontal cortex and then even more the motor cortex seem to have more and more recurrence, more and more internal activation, and less relying on external input. So the working point depends on cortical area. Now I have 2 more slides. I think I very quickly go through that. And these basically ask, so why should there be different working points? Why shouldn't one be, remember this first slide, why shouldn't one always be at maximal information transfer? And we argue that for many tasks, you don't need maximized information transfer. To investigate that, we went to investigate in a neuromorphic chip. They resemble human neocortex very roughly. It's spiking integrated fire neurons, excitatory and inhibitory ones. These recurrent networks on a neuromorphic chip are in principle Turing universals. They could do anything. They distribute processing. Running and training doesn't scale well on an ordinary computer, but on these neuromorphic chips, it does. We implement local learning rules, in particular a homeostatic rule. And then we can either take the statistical physics perspective, or on this neuromorphic chip, we collaborate here with the Heidelberg group, and we can apply that. I will skip this one part, but I want to go here now to task performance. Let's take first an abstract view on it. So what we find is if we have now weak input and thereby enforce strong recurrence because homeostasis brings the network to a certain spike rate. So for weak input and strong recurrence within the network, you also find that the mutual information between neurons, the transfer of information between neurons, and also the storage within a single neuron, how much they can read out, both are higher the weaker the input and the more recurrence you have in a network. But this is abstract, this is information theory. What about real task performance? Yeah, we took a simpler task, which is just summing, or a more complicated task, which is a parity task. It's a fully nonlinear task. You find that the more complicated task, this profits from more internal recurrence, whereas the simple task profits from listening more to the external input. So this is absolutely coherent with what we see in cortex. The sensory areas might be more listening to the outside. They have more input and less recurrence relative to frontal areas that seem to have weaker input and more recurrence. With this, I want to summarize. I started with quantifying spreading dynamics and the relation between the exogenous and endogenous activity. I showed you that the working point might really depend, or it seems to depend on cortical area. And this is coherent with the idea of task performance. That task performance seems to be really not the question of there's one optimal working point in terms of recurrence, but depending on your task requirements, you might need really completely different working points. This I would really like to thank my group. Looking forward to the discussion later.
Viola, thank you very much. Another excellent presentation and really, really interesting. You've pointed out the complexity of the biological systems and how it's challenging to apply usual rules of physics to them, but then shown how you can develop models which can then be very useful in prediction, and again how you bring in real world data to sort of validate those models as well. So thank you very much for that. I think it's a perfect example of why we're having a workshop like this, to bring the application of physics to some of the biological problems. So thank you. I'm sure we'll come back to some of these things in the panel discussion. So it's a pleasure to welcome our final speaker of this second session, who's Neil Oxtoby. Neil is a UKRI Future Leaders Fellow at UCL in London, where he's attached to the Department of Computer Science and Engineering Science. And he's also data science lead for the Deep Dementia Phenotyping, or DEMON network. I'm sure Neil won't mind me saying his webpage says he's a scientist, a musician, a data analyst, and a computer programmer. So lots of very, very important skills to have. And his research is in computational models that enhance our understanding of degenerative disease. And today, it's a pleasure to welcome him to talk about top-down and bottom-up computational models for Alzheimer's disease. Over to you, Neil.
Yeah, I used to be a physicist, and now sort of calling myself an Alzheimer's researcher. But in the POND group, Progression of Neurodegenerative Disease, at the Centre for Medical Image Computing at UCL in London, we work on lots of different neurodegenerative diseases. So what I'm going to tell you today is applicable to other conditions beyond Alzheimer's as well. Thanks again for the invitation. So I took a kind of bold approach and promised a title before thinking about it too much. It turned out to be quite a fun exercise in putting this talk together. So the reason I'm here is what I do in the POND group and what we do in the POND group, what we call data-driven disease progression modeling, which is attempting to fuse snippets of multimodal data from multiple individuals into quantitative signatures of disease progression. So these trajectories that Zoe mentioned earlier. And I'm going to talk about two different kinds: top-down models, which basically work on phenomenology, what we can observe, and very much motivated by something that James brought up earlier, precision medicine from the phenotype perspective, and bottom-up models, which are our attempts at modeling disease mechanisms. And here's a semi-famous example from a recent paper that came from our group, a very collaborative work with some others on subtyping Alzheimer's disease. So my talk will have a neuroimaging lean, and Heidi gave a great talk earlier on different modalities of neuroimaging that can be useful in this space. but that's because I come from the Centre for Medical Image Computing. But we're very motivated by clinical impact. We work very closely with our clinical colleagues at the Dementia Research Centre at Queen Square in London. So the sort of bold concept I had when I proposed my title for this invited talk about top-down and bottom-up approaches is about generally understanding complex systems, and brains are about the most complex systems we have access to, and brain diseases are even less understood due to this complexity. Top-down approaches. Hello? Top-down approaches work on phenomenology. So in vivo data, clinical data, things we can observe in vivo, and are quite amenable to data-driven approaches to discover patterns and characterize these progressions, but give us less mechanistic insight. It's kind of like reverse engineering when you're trying to understand disease mechanisms using top-down approaches. Bottom-up approaches almost need the mechanistic insight to start with, but if they work, if they fit well to the data, then you can understand the disease mechanisms from observations. At the moment, it kind of requires fairly invasive methods to really understand what's going on, and the data-driven capabilities and aspects are a little less known. This is sort of forward engineering. So this is kind of what I was thinking when I proposed the talk title. This was mentioned earlier, actually, by a couple of the sort of speakers about what we know about Alzheimer's disease, in that it is defined by post-mortem histopathology and Braak staging. And it takes a long, long time. There's a long pre-symptomatic period before the clinical syndrome kicks in, and memory being the most famous one in Alzheimer's, but there are many others, including visual, etc. So there's decades of pathology happening before we can actually observe what's going on, except you can access this pre-symptomatic period in some rare familial or inherited forms of Alzheimer's and other neurodegenerative diseases. We know a little bit about risk factors from genetics, and Henrik mentioned a few of these things, but we don't really understand too much, and we can't enroll enough people in the study for long enough and keep them all in the study from early enough and do enough invasive measurements and non-invasive measurements to just observe what happens to understand the disease progression. So we need to be more clever about it. And what's added to this to make it more difficult to understand Alzheimer's and Alzheimer's progression is the heterogeneity in both the syndrome I mentioned earlier, the onset, so the disease timing of onset of symptoms, the progression, speed of progression, and exactly the details of the progression. And even in the pathology underlying all of this. The good news is that we can probe some of this pathology in vivo, which was mentioned earlier today. Now, this heterogeneity causes all sorts of issues for understanding the disease, but also is a large contributor to the reason why we don't have treatments for Alzheimer's. And Craig Ritchie mentioned this a little bit earlier. That most of the treatments, pharmacological treatments for Alzheimer's, are based on this amyloid cascade hypothesis, where amyloid abnormality comes first. But some people have been questioning it since we've had 2 decades worth of failed trials, basically. Multiple reasons why these trials might fail, and Craig mentioned a few of those. Maybe we're intervening too late. We need to be thinking about prevention rather than cure. Maybe we're getting the wrong people in, so they could be due to this heterogeneity and due to the disease staging. We're looking at symptoms too late. The trials may need to run for longer. We may need to, these amyloid, for example, may need to be treated for longer to have a lasting effect and a measurable effect in the clinic. The insensitive endpoints, again, was brought up earlier about the difference between having biological markers as the endpoint of the trial rather than just a clinical benefit, which is obviously important to the patients and the families and shouldn't be disregarded. And the amyloid hypothesis may be wrong, or just inadequate, not quite complete. Comorbidities are a big problem. Maybe we need multi-target strategies. What has and can be done about it is what I'm aiming to talk about from the point of view of the computational models that we develop in POND. There's a couple of recent papers of ours that talk about relevant things on this slide. So, top-down models. I'll tell you a bit about the journey on the road towards what we call data-driven disease progression modeling. Traditional models back a couple of decades ago sort of assigned a disease stage and measured disease stage by symptoms. So you basically use regression, and here's some maps of neuronal atrophy in groups of patients, pre-symptomatic, mild and moderate Alzheimer's patients, subdivided by general cognition test score, the MMSE. In familial Alzheimer's, as I mentioned, you have a time axis against which you can regress, and you see different biomarkers diverging from controls in patients, in preclinical patients, pre-symptomatic patients that you know are going to develop Alzheimer's due to one of the family of mutations that they've inherited from their parents. And you can map the disease a little easier in these rare forms, but you can't use regression, generally speaking, in sporadic forms of Alzheimer's. What you can do is use some pattern recognition once you've got enough data. So here's some 2008, 2011 results on the left, some classification results similar to what Zoe started talking about in her talk for differential diagnosis between Alzheimer's and frontotemporal dementia, as well as Alzheimer's and controls, with a heat map from the image, the neuroimage of the brain, showing you the informative areas. And this can be extended beyond neuroimaging into the disease state fingerprint of Thiele et al. That has also gives you weights for the cognitive scores, the imaging and the non-imaging biomarkers contribution to the classification power of this classification model. Beyond that, 2004, sometime in the middle of that actually, the ADNI initiative started, and that's, I can't understate how important this has been, the influence of this particular study and others like it that have often modeled themselves on ADNI at enabling people like myself and other machine learning folk to be able to unleash the power of computer science on neurodegenerative disease. And this led to the hypothetical model, Jack curves of Alzheimer's progression, showing a cartoon. These aren't actually trajectories. The hypothetical model is actually the sequence of changes here from clinically normal asymptomatic through mild symptoms into dementia, which was updated with a detection threshold a little later, and a little bit more details about some more recent biomarkers that were available. And then thanks to Laura's talk earlier, there is now also the vascular component that's been added that I'd forgotten about while preparing my slides, but managed to get it in. So the hypothetical models are what really inspired the data-driven approaches. Computer scientists saw these hypothetical models and said, oh, we can do that, there's data. ADNI's been around, for example. What can we do? Well, ultimately, this variability that I mentioned in onset and progression means that we, as a general class of methods, we implement, design, and fit pseudo-time methods. So methods that allow to estimate your stage of disease along your trajectory or a group level trajectory. And there's a couple of different kinds. There's a discrete one called event-based modeling that came out of the lab that I joined back in 2012. It came out about the same time I joined the POND group. And there's a couple of papers there. This is from the Young paper in 2014. It's the event-based model of progression showing early CSF markers, atrophy, cognition, and brain volume changes. A dark diagonal on this positional variance diagram would be a very strong confidence in the ordering. So this is just a discrete model of disease progression. There's no real sense of quantifying time, but you do get a pseudo time score or a disease progression severity score. And when you bin, this is the ADNI data again, when you bin the cognitive categories, you see Alzheimer's patients in pink, mostly later in the disease, having accrued more abnormality across this suite of biomarkers, and the cognitively normal group mostly towards the start. And this disease stage was useful in this paper in 2014, was shown to be useful for survival models for predicting conversion from MCI into Alzheimer's disease. There are continuous versions of these as well. So these pseudo-time methods, broadly latent time methods, like the latent time joint mix model of coming out of Mike Donohue's lab, item response theory and Gaussian progression modeling, Gaussian process progression modeling, really nicely Bayesian probabilistic estimates of group average trajectories, data-driven Jack curves, if you will. And these also give you a pseudo-time that correlates nicely with cognitive status from blue through to red, progressing in Alzheimer's disease clinical status, has an increasing disease stage. What we've done with our sustain algorithm, subtype and stage inference, developed by Alexandra Young, who was a PhD student and postdoc in POND, recently was added clustering on top of the pseudo-time disease progression modeling. And this is a picture of the abnormality estimated from cross-sectional data in 3 automatically detected clusters of Alzheimer's disease progression within the ADNI data set. And this has been extended. This has the pseudo-time sort of element, the staging element, the really fine-grained staging ability, but also added in some clustering. So you can have typical cortical or subcortical Alzheimer's type, and that may have different outcomes for patient management decisions, etc. And this has recently been run on a very large tau PET dataset by Jake Vogel and colleagues from a few datasets from Biofinder, ADNI, UCSF dataset, and a South Korean dataset on a different tau PET tracer. So they found 4 subtypes actually, which were quite common, all had roughly equal proportions of individuals assigned to them, and was robust across traces as well. That's available on medRxiv, should be out in a journal very soon. So this is a sort of whirlwind tour of the top-down approaches to disease progression modeling. What does it tell us about disease mechanisms? And this is important for, potentially important for developing new treatments if that's what's needed to fix this clinical trial problem in Alzheimer's disease. So can we understand and explain top-down observations of pathology, like from neuroimaging, using bottom-up models of mechanism? And this sort of started around the same time, back in 2009, with ideas about selective vulnerability of brain regions and atrophy patterns being explainable based on a network and a few different hypotheses of what happens for the pathology that spreads, like amyloid and tau. And it could be, the brain could atrophy based on wear and tear, based on proximity to where the pathology starts, where the proteins start to misfold, or the regions that don't get used. And these hypotheses can come up with predictions, and those predictions can be tested. So this led to the spreading models, basically, the prion hypothesis of Alzheimer's disease and other neurodegenerative diseases. And this is where physics starts to step up. So the network diffusion model, which is essentially a heat equation on a graph built on the anatomical connections in the brain, estimated from magnetic resonance imaging and tractography, and an epidemic spreading model, which is more from epidemiology. So the idea is here that connectivity in the brain can predict pathology that we can measure. And this gives us some information on the underlying mechanisms. So for example, the epidemic spreading model is quite good at predicting amyloid spread, in this case, across the brain. More recently, some actual physicists got involved in the network spreading idea, and they added on top of this global brain connectivity spreading, some local kinetics, like misfolding of proteins, the actual prion process of encouraging extra misfolding, clearance of these things, and these models increasing in complexity and the number of parameters that you need to fit, and the challenges for finding the kind of data that is required to estimate all these parameters is increasing here. And these are 3 different models. A couple of key references there, and there's a more recent paper came out last year where they looked at the simplest version, the Fisher-Kolmogorov, and attempted to fit the parameters in that model. So these are what I'm calling bottom-up models, and the most complex one has, you can see all these parameters, and all these parameters would apply, if you're thinking about it in terms of neuroimaging, all these parameters would apply in each voxel or each pixel of that 3D image in the brain. You can see how complex it is to try to fit these models using the data that we can get from the top down in the clinic. There's also mouse model work, and I won't talk too much about this, partly in the interest of time, partly because I've only just started really getting into it. But here's a paper, a fairly recent paper, that looked at network diffusion plus selective vulnerability. So some of the ideas I briefly discussed just before, and showed some good correlations in predicting pathology, so predictions on the horizontal axis, and measured pathology on the vertical axis in mice. So wrapping up, a bit of a whirlwind tour of what I call data-driven disease progression modeling, which is sort of statistical modeling, some of it physics, inspired by physics, some of it directly from physics. That is a growing field where across the world, actually, with lots of different groups involved. We've had recent conferences dedicated specifically to this kind of approaches. And the take-home message is that I think the physics of life and the physics of disease modeling is super important. These computational models, inspired by, based on physics, are indeed improving our understanding and clinical management of neurodegenerative diseases at multiple scales, with all the challenges that I mentioned, and probably more between linking the 2 scales. A quick overview of some of the related work from Pond, a former postdoc of ours, Sarah, has a nice paper out in eLife. She worked very closely with Marco, who's also a former postdoc of ours. on topological progression profiles, so brain connectivity and disease progression modeling, these pseudo-time approaches, and how they differ in aging, Alzheimer's, and multiple sclerosis, good for differential diagnosis as well as disease understanding. And as a PhD student, working more on fundamental things related to tractography, which is how we estimate these brain connections, these connectomes, and then use those in our mechanistic bottom-up models. Isaac's a very recently started PhD student of mine, and he's very much aligned with this workshop in that we have a bold idea to try to link these two scales, the bottom up and the top down approaches. So we've enlisted Mark Bush at the UK DRI over at UCL as a co-supervisor of Isaac's PhD to sort of try to do these kind of things, which I noticed from Paul's opening talk this morning. This lines up with his conceptual challenge number 4. Actually, so do the two former examples. Hanyi is a postdoc who recently joined us, working with Andre Altman in the Combine Lab, the Imaging Genetics Lab at UCL CMIC in the center. And they're both involved in, as am I, the EDADS project, which is more about early detection, trying to marry disease progression in patients and the modeling through to as early as we can midlife risk factors using various things from computer science, which aligns with Paul's conceptual challenge 1 about distinguishing everything early. And my own fellowship with the postdoc Cameron Shand, where we're looking at trying to individualize AI, machine learning, disease progression modeling for medicine with Alzheimer's as the primary focus, and looking explicitly at how we can benefit clinical trials for the top-down things, as well as understand disease mechanisms, so the bottom-up modeling that I've mentioned. And anyone interested in the challenge of forecasting Alzheimer's disease, thoroughly recommend that you check out the Tadpole challenge that I was a co-organizer of. There's a couple of publications here, pre-publications here you can check out. Lots of submissions to that from data scientists through to researchers. Even some high school students got involved to try to forecast clinically relevant Alzheimer's disease outcomes using ADNI data. So this was part of the EuroPond consortium, which is led by the Pond group at UCL. And we had some generous contributions from these 3 charities for the prize money for the initial phase of this challenge, which is over. And this second paper here is the result paper. But the submission remains open if you want to submit your favorite forecasting model to predict outcomes in individuals at risk of Alzheimer's disease. Uniquely, we didn't do this in a sort of cross-validation setting. We didn't hold out any data. The deadline for submission was before ADNI 3 data was acquired. So this was a genuine prospective forecasting challenge. And the acknowledgement slide. If you want any more information, go and check out these websites for the 2 consortia that the Pond Group leads, the Pond Group itself, some more details in there and contact details. And our collaborators are wonderful, especially at the DRC, but we collaborate more widely than that. Data providers who make my job even possible, and the volunteers who make the data providers' job possible as well, patients and families involved in all these studies. Extremely valuable stuff and much appreciated contributions to what we're trying to achieve, which is essentially this phenotype precision medicine concept was mentioned earlier. And in my heart as a former physicist, I'm very interested in bringing the power of physics and computer science to bear on this super important problem.
Thanks very much, Neil. That's great to hear about the different granularity that the top-down and bottom-up approaches can give, and also about Pond, EuroPond, and Tadpole. There's a bit of a theme there, isn't there? I'm not sure if it's a top-down or a bottom-up theme, but great to hear about that.
It was very top-down. Very
top-down. Can I now invite the other speakers from session 2, both before and after lunch, to join us, and also Carl, of course, as co-chair. And please do send some questions in the Q&A. I see there are a couple, but please send some more. And maybe I could just kick things off while people are thinking and putting questions in, because it was mentioned a few times, particularly in the afternoon talks. So Zoe and Neil, you mentioned it about sort of lack of data. And I just wanted to explore that a little bit more about what types of data you thought were lacking and would be needed, because we have quite a lot of data, and John talked about DPUK2, and you mentioned ADNI, and there's ABLE, and there's Prevent and EPAD we heard about, and Biobank. So there's quite a lot out there, and I know modelers, you might well say the more data the better, which I totally understand. But from your point of view, is there any particular types of data? Is it more data? Is it more frequent assessments? Is it over a longer time period? You know, different diseases. What sort of data do you feel is lacking? What would be most helpful? I don't know if, I mean, Zoe, do you want to?
Yeah, sure. Maybe I'll kick off. So I think longitudinal data is really very important. As Neil also mentioned, he said the trajectory modeling approaches are really key for early prediction at pre-symptomatic preclinical stages, and longitudinal data is really important. I think also it'd be really great to see digital data being added together with biomarkers, blood marker, obviously data as we are coming along, and digital data to combine with the gold standard imaging data that we currently have quite plenty. But I think, John, another issue that comes to mind is accessibility to this data. We have made a long way forward, especially with BP UK efforts, but still we got some way to go to be able to share data openly and be able to, invite challenges like Neil did with the Dartboard challenge. I think this is really important for the community. We will see ourselves building models that are really efficient very quickly and can be more faster and more easily transferred into clinical practice.
Okay, thanks. Anyone else, Neil?
I think the general answer to your really fascinating question about what data is needed, it comes down to the specific question. I'd love to have more data from these failed clinical trials, but it's hard to prize from the hands of pharma often for various reasons, practical and others. But for the mechanistic models, if you're trying to understand what's happening at the neuron level, having more PET scans probably won't help. So it comes down to the specific question, I think, as for what data is needed.
Okay, thanks, Neil. John, did you want to come in?
Yeah, I think that if you're looking at it from a mechanistic perspective, I agree with what's been said. If you're looking at it from a more general perspective, the hardest data to access is actually electronic health record data. It's amazing to me that the mission of some institutions seems to be not to give data as their raison d'etre. And these data are incredibly valuable. They would inform our research cohort data many times. They would provide a basis for testing your laboratory tested hypotheses in the public realm as surrogates, as Paul Matthews said earlier. So I find there's no good reason for this. The UK has some of the best data in the world. If only we could get our hands on it.
Okay.
If I can say, John, something that I realized when I was trying to sort of build a trial around essentially disease modification in diabetes, so specifically on dementia. And what I realized that really, although we know that certain conditions associated with risk with dementia very strongly, we do not necessarily know what the interaction is in the preclinical stages. The thing what may be required is that we have dedicated dementia cohorts based on one strong risk factor like diabetes, and then monitor these people or chronic kidney disease or hypertension, because most of the data is coming cross-sectionally, and we lack the granularity, for example, of diabetes control, of hypertension control, to be able to inform trials in these populations. And I think that may be the key, actually.
If I could just build on that, the most population-based data comes from, if you like, generic cohorts. Now, these cohorts have been specifically designed to address multiple questions. And that's, it's extremely efficient. Let's not pretend otherwise. But when you want to address one question very explicitly, the data are usually inadequate. So, for example, to find population-based data on disease progression for neurodegeneration is extremely difficult because the cohorts were not designed to show that. And I think we're entering a period now where it is quite legitimate to say we do need some disease-specific, pathology-specific, or syndrome-specific cohorts which have assessments designed to be standard over time that will inform disease progression from the basic cellular beginnings through to clinical outcomes and through to death. And that means integrating not just research measures, population-based research measures, but those must then walk into clinical measures and then walk into registries, ONS and things like that. Now, it's completely doable, but it hasn't been done.
Do you think, this is Henrik, do you think we should try to get together and sort of nominate cohorts for, I mean, what we would like to contribute would be actually the blood biomarkers I mentioned, and we are now trying to increase our throughput in the cruciform lab by building something we tentatively call the biomarker factory. So we would try to scale up everything I showed in my talk. We tried to scale it up so we could start to plan for doing tens of thousands of samples instead. But of course, then we would need to collaborate with all you guys to know what would be reasonable to, if one should nominate sample collections, whether would be good clinical end points and perhaps some other interesting results, perhaps genetic data, where we could formulate some hypothesis where this would make really good sense to do that type of large scale, also blood biomarker work.
Yeah. Henrik, I think it would, and I think it's a very time-efficient way of doing it. Now, typically, when cohorts are approached to do this, one investigator approaches one cohort and says, "Can I have your 300 very valuable samples?" And of course, the scientific case is weak, and therefore it's declined. However, if there were a group of people who wanted to get together and strategically say, we will conduct a feasibility analysis, these are the cohorts, these are the numbers of samples, this is probably the best use of these samples that you're going to achieve in the next 10 years, then I think the prospect of success would be high.
And John, can I just clarify, are you thinking in terms of existing cohorts, or are you calling for a new dementia-specific bespoke cohort?
So Henrik's suggestion was for existing cohorts because it's very time efficient. My view is that in addition to that, we need to collect data prospectively using standardized tests that will carry on through into clinical practice so that we can go from, if you like, the inception of disease through to diagnosis, through to death. Now, we can do this. The time is right for doing this. But when I've approached various pharma companies, etc., they've been reluctant to invest.
I totally agree with you. I also think it's needed. And as someone who's designed several cohort studies, you simply can't do everything. The burden gets too much. So you need to focus on your area of interest. And I think we know these studies are expensive to do and require long-term commitment to undertake them, and that's really, really important as well. And we've got a funders session coming up later, and it might be interesting to get comments on that, because that's one of the challenges, very real one, of going back every 3 to 5 years for funding, as we all know, I think.
Yeah, John, I think with modern technologies we can do it much more cheaply. You know, if you're not measuring, you know, if you're not boiling the ocean, then you don't have to pay for it. If your measures are focused, and you're using digital technologies remotely. The critical thing is getting the biomarkers efficiently collected and processed properly. But I really think it's completely doable, and I would love to hear the funders' views on this.
Okay, that's one for a little bit later. Carl, do you want to take anything that's come in the Q&A?
Just a couple of, I thought, quite interesting exchanges in the Q&A, a little bit sort of focused, but I think nice to give the opportunity to Henrik and Laura just to revisit some specific questions about the blood-brain barrier, endothelial biomarkers and the like. I thought that was informative. I was wondering if the two of you just like to rehearse your answers and pick up the interesting points that were raised for the rest of the audience.
Yeah, so Axel raised the question about whether water exchange was measuring actually quite a fundamental different process to looking at gadolinium leakage. And I think I tend to agree. I mean, there's many more pathways that water can exchange. And so maybe it's not that that measure is more sensitive, it's just measuring a different process. But nevertheless, it's picking up something which, a process which is altered in cognitive impairment, which can't be detected with gadolinium leakage. So I think that was the key point there.
And I got a question on fluid biomarkers for well, integrity of the neurovascular unit, and also endothelial biomarkers. And what I mentioned in my written reply there was that there are exciting results on VCAM and ICAM related molecules in blood, but it's hard to tease out what reflects cerebrovascular changes and just general vascular changes. In CSF, there is one interesting biomarker that came from the Slokovich group in California, the soluble PDGF receptor beta biomarker. So PDGF receptor beta is really a parasite marker. It turns out that when parasites are stressed or part of a leaky blood-brain barrier, then they secrete more of the soluble form. They had a very nice study where they compared some MRI measures of blood-brain barrier leakage and CSF levels of soluble PDGF receptor beta. This class of biomarkers to me has become so interesting from a neuro-COVID perspective, because there is certainly something going on there in many of many people who have had COVID with some, well, with quite with a lot of CNS symptoms. It really looks like there is something going on in the endothelial and parasitic compartments in the brain, which also is reflected then by neurofilament light increase in plasma in those who have had more severe forms of this disease. Or syndrome, I should say, probably. But actually, my answer was that there are no well-established biomarkers that can tell if the cerebrovasculature specifically is changed. But CSF soluble PDGF receptor beta is an interesting one.
Yeah, thank you. I mean, it's a particularly prescient issue with long COVID and potential long-term neurodegenerative consequences, especially say for children. This is a very active debate at the moment about whether we should prioritize the vaccination of children. So it's absolutely fascinating.
With Paul, we will do a study now on long COVID with Paul Matthews, with blood samples blood and imaging measures collected before and after COVID, and he's selecting people who have had more severe COVID. That will be an exciting. It's almost like a natural experiment.
Uh-huh. These conversations really speak to the sort of the sort of key mechanisms of pathogenicity and our understanding of that. And I sort of want to open up the discussion. And I have a question for all the panelists, and in fact, my co-chair John as well. And the question is really, what has physics got to offer? And if it has anything to offer, are you already doing the physics that you require? And I'm going to frame that question just by coming back to the earlier theme, which was this nice distinction that Neil brought to the table between the sort of top down, the bottom up. In my world, this would be the difference between a sort of forward or a generative model versus a predictive or classification model or a recognition model. And it strikes me that the physicist would probably get more interested in the forward model. So I'm thinking Viola's presentation, for example, a percolation model as a way of generating the kind of data that we have access to and understanding what's under the hood in terms of the connectivity and the message passing. So I just wanted, if possible, just to go round and ask everybody whether they thought that we should all commit to a data-led top-down approach and throw a lot of machine learning at the problem, or whether that's the wrong thing to do, and what we should really want to drill down on are the good generative compartmental causal models, epidemiological models with well-defined, plausibly interpretable factors that generate the data, and then try and invert those data, or both. And if both, how you make them meet, and if that's the right endeavor, or one or two or three of those are the right ways to approach. Is there anybody in physics that you would turn to for help? And that's a bit of a cheeky question. I imagine Neil and Viola already think they are physicists, but you know, there may be other kinds of physics out there that would really facilitate this game. So does anybody want to take that? I might pressure John, in fact, John O'Brien, to see what he thinks, because he's been courteously asking everybody else's views so far.
Well, thanks, Carl, for that. I mean, the answer, of course, is both, isn't it? But you know, you said where could physics, you know, have the greatest input, or what's missing? And I suppose, coming from the imaging point of view, I see that we're using these approaches, as we've been described, in bringing different modalities together. You know, getting data from the different modalities in the scans and then putting them into an algorithm, machine learning, AI, whatever. And that's very useful, and that's absolutely fantastic. And that's producing, as we've seen, some great results. But I'm not sure we're so good at actually integrating that data in sort of imaging space in a way, in a multi-dimensional way, that might actually prove very valuable. And I think that's where maths and physics might be able to offer. And that's a bit of top down and bottom up, I think, depending on which perspective. So that's where my wish would go in terms of bringing physics and maths into imaging, at least. And there's no reason why we need to limit it to imaging. There's biomarkers and other things from blood and CSF that could be brought into that.
I would echo that, in the sense that it's all very well having a fairly sophisticated data platform like DeepUK, but the development of that platform is driven by the use cases. And to have the physicists and the clever mathematicians come in and say, okay, this is the computational facility, the computational architecture we need, then we can build it for them. So I think that would be very, very helpful.
And I need physicist help when we work on more sensitive assays. That's really, with some of the technologies we are working on now, it's about single molecule detection, and their physics play in a lot, and also the possibility of multiplexing with different types of signal generators. I think we could do a lot of exciting things together in that particular field, to be able to measure multiple analytes, single molecule level, and with different types of fluorophores or other potential signal generators.
Yeah, I would agree with that perspective. I think just basic measurement science that physics brings is critical to a lot of the imaging measures we develop in the brain. And I think I'd also make a slight ploy towards the bottom-up approach. I think there's nothing you can do better than having a very clear question, and you're using scientific methods to test the hypotheses you have.
I'll pick up on Laura's point. I think physics is super valuable, as everybody said, in building kind of more sensitive tools for us and extracting information. So there's more kind of sensitive markers can only improve our ability to make clinical predictions. And then I think where the maths and the computational science comes in is really in harnessing this power and looking at interactive relationships between these more highly sensitive features that we can extract through more kind of elaborate and advanced physics measurements. But the beauty, I think, of bottom-up approaches is that they look at the data with no assumptions. Hypothesis is obviously really great, but sometimes we miss out things that we wouldn't have seen in the data. And the more sensitive our physics approaches, the more we can see in the data with computational approaches. So the two go really both hand in hand. And I think Steve's comment in the chat is also kind of posing the challenge of how do we link this, especially when we look at different time scales. So it's a great challenge.
I mean, physics is a huge field. I think it's absolutely clear that developing new techniques is fundamental. It brings new microscopes, it brings fMRI scanners and so on. But then more about the other part goes a lot into the direction of data science, AI for the exploratory direction. What I really think is a strength on physics is to think about a potential but still abstract mechanistic relation. And what I at least try to do is having the mechanistic interactions in a way that I can solve it analytically. The advantage of having an analytical solution is that I know every single parameter and I know this model. If I then have, let's say, more the computational approach from a very complex model, sure, I can tweak a parameter, but I don't know what's really the effect of it in this highly complex model, which is complementary, but it's a more exploratory approach, almost experimental. To add something to where I see a lot of promise, often you don't know where the promise is coming, and we don't know what's the breakthrough in 50 years. Who would have foreseen the deep learning so clearly in the 70s? For good reason, there was an AI winter. So it's very hard to predict. What I see is very interesting at the moment is the neuromorphic chips. It's something that's also in the physics domain because it's semiconductors on the one hand, and it is now putting, and I think really opening the potential to bring recurrent networks and local learning rules to a new power. It's not about applying it directly to the brain, but it's simply because of computation. The size of a theoretical model or computational model with local learning rules that you can model on conventional hardware is really limited. If you want to update your synapses regularly, no way you do that. And the neuromorphic chips will be able, I think, to scale that up. So I'm very excited about that direction, but sure, it's also a bet to set on to go in that direction.
Are you proposing them giving neuromorphic chips Alzheimer's disease and sort of having little physics engines for us to generate synthetic data? And
yes, absolutely. I mean, it would be more mechanistic in the sense that one would have to know which parameters on these neurons do I now have to change, so that potentially something like Alzheimer emerges. But at the moment, these chips are still small, no matter where you look. Either they are graded still or it's spiking, but let's say I think Heidelberg has now 500 neurons. It's still fairly small, but I think it's a question of scaling up. It's not a question anymore of new technology. It's all there. It's only a question of a bit more money and scaling this up.
If one could model your networks with where you have plaques popping up and causing small network disturbances, and perhaps also how networks can compensate for that, and by that potentially model the preclinical part of amyloid pathology, where I mean, sometimes we wrote in review papers that amyloid has a silent incubation time, but it doesn't make sense from a biological standpoint. And now when we have learned that amyloid will immediately cause tau phosphorylation release, but still the network seem to be able to cope until a certain, until there is too much damage evoked.
That's something one could beautifully explore. I mean, these chips run by a factor of 1,000 faster than a biological brain, so we don't have a problem with also aging these systems. And one could, in principle, see whether there is, damages that can be compensated. You hardly see them, but at some point the compensation doesn't work anymore, and then you see it in the cognitive decline. That's something that would be very interesting to explore on such type of networks.
So I'm really sorry to break up this fascinating panel discussion, which is getting very lively, but we do need to move to the next session. Just very briefly, though, because there was a question about taking part in research, and clearly none of this would be possible without research. And we heard about registers early, and Join Dementia Research is the way to get involved in dementia research and to find out about studies. It's a register run by the Clinical Research Network. You just type it into your favorite search engine. You can sign up, find out about studies there. So please do that and signpost people to that. Can I just thank all the speakers in the session today, and Carl obviously as co-chair. It was fantastic talks, really interesting panel discussion. Thank you very much. And Steve, over to you for the next session.